So good afternoon, everyone. Welcome to this session, educational course organized by the International Eczema Council. This is our second course in the AAAAI. The previous one was 2 years ago. I'm Ioana Agace. I will be the moderator of this session today. I'm an allergist working in Europe, Romania. I was the president of the European Academy of Allergy and Clinical Immunology, and currently I'm a counselor of the International Eczema Council. I would like to express my thanks also to my co-chair, Dr. Jeff Yu, who could not be with us today, but who put a lot of effort in organizing this program. We would like to be as interactive as possible, and we try to cover all the aspects of environmental influences on atopic dermatitis, trying to make a good mix of basic and clinical science and also pediatric and adult cases. So these are some housekeeping rules. So please, no recording all the session will be recorded for you and available on the International Eczema Council website. Of course, silence your phone. Use the mobile app of the meeting for Q&A, but we can also have questions live from the room. And also go into the session, engage, and let's try to make it as interactive as possible. Now we will start with a brief introduction on the International Eczema Council presented by its president, Dr. Ellen Irvine. Dr. Ellen Irvine has been a consultant dermatologist at Children's Health Ireland and St. James Hospital in Dublin, Ireland since 2002, and professor of dermatology at Trinity College Dublin since 2007. He has published extensively in high-impact journals such as New England Journal of Medicine, Lancet, Nature Genetics, and Nature Reviews Disease Primers, mostly on epithelial genetics, characterization of skin barrier, disease mechanism, and therapy of atopic dermatitis. His international awards for research include the Times Higher Education Supplement Research Project of the Year and the Paul Gerson-Una Prize, brought by the German Dermatology Society. He has delivered several prize lectureships, including the MB Sulzberg, the Watson-Smith, the Dolovich, the AAAAI. Dr. Irvine is a founding director of the International Eczema Council, an elected member of the Royal Irish Academy and the Académie Européenne, and a honorary member of Société Française de Dermatologie. He will address us virtually today because he could not be here.
So it's my job to welcome you to this IEC symposium called The Impact of Environmental, Food and Contact Allergies on Atopic Dermatitis. We're delighted to partner with the AAAAI and the WAO at this Congress, and I hope you find this next session informative and educational. Just a few housekeeping comments about this session. It'll be recorded and it'll be on demand on the IEC website and also on our YouTube channel. If you have any questions, there's an email that you can contact here. If you're interested in joining the IEC, or if you have any questions about what we do, our mission or our purpose, please contact Alan Stiles at the email here. Briefly about the IEC is celebrating its 10th anniversary this year. We are now in 33 countries. We have 151 counsellors and associates across 6 global regions. We grow every year in adding new countries, adding new counsellors, striving for diversity right across the board. We have 117 counsellors. These are well-established clinicians or researchers in atopic dermatitis management. They have multiple peer-reviewed articles and actively engaged in leadership, clinical trials, and scholarship. The 34 associates are early career clinicians or researchers who have finished their residency training, who are fully qualified specialists, and they've published some early articles on atopic dermatitis and already are making meaningful contributions. And we're keen to add more allergists to our numbers. Our articles were changed a couple of years ago, and this, just to serve this notice that we're keen to have people who come from an allergist or an allergologist background, as our counsellors or associates, as we've previously mentioned. So do consider joining us and contact Alan Stiles at the email address given. We've expanded our eligibility. I've mentioned that we have dermatologists and allergists, but now in 2025, for the first time, we are taking PhD researchers. These are researchers who are not clinically qualified, but who have a track record of investigations, meaningful investigations and publications, and who've made a contribution to atopic dermatitis understanding. And our first PhD counsellors will be formally admitted in the first quarter of 2025. Our mission is to improve patient care by promoting research, education, and best practices in treatment for atopic dermatitis in people worldwide. Our vision is to transform the lives of people with atopic dermatitis. Our anthem, we're a global community of leaders in atopic dermatitis research, treatment, and prevention. And our values, we're patient-centered, innovative, evidence-based, globally inclusive, collaborative, and forward-looking. And if this is something that you think you'd like to be involved in, working with a great bunch of people, please do consider it. We are very grateful to our sponsors at President's Council, Executive Council, Directors Council, and Councillors Council level. These are hugely important to our ongoing educational and research activities, and we wouldn't be able to do these without the support of everyone listed here. Our symposia this year, we have our first 5 symposia locked and loaded. Obviously, today is the Environmental, Food and Contact Allergies and Atopic Derm in San Diego. Our next meeting will be at the AAAAI, and that's Converging Pathways, Atopic Dermatitis and Alopecia Areata. We'll have a number of patients, and also the National Alopecia Areata Foundation and the National Eczema Association will be there. So that's a really interesting meeting too. The World Congress of Pediatric Dermatology is in Buenos Aires in Argentina, and that'll be a session on strategies for preventing, treating, and managing pediatric atopic dermatitis. That's in April. Shortly followed by that, in May, we're back in San Diego, and this is for practical applications of multi-omics as at the Society for Investigative Dermatology. That's a very PhD-friendly, research-intense meeting. EADV is in Paris, France, in September 28. And that's talking about the relationship between atopic dermatitis and cutaneous T-cell lymphoma. That's a dynamic space, a lot of new information coming out, and some important clinical questions. Please do find and connect us with us on whichever platform you usually use. We've got Facebook, X, LinkedIn, and YouTube channels. We want to grow our interactions through these social media platforms, and we're very keen to hear from you and for you to connect with us through this. Our YouTube Learning Centre has over 70 hours of content. It's been viewed over 10,000 times so far, and there's multiple lectures on every imaginable topic on atopic dermatitis, and including today's symposium will be available to watch back in due course when it's edited, curated, and posted. Any questions about IEC or interested in becoming a sponsor or a counsellor, you've got Alan's contact number here. Alan's our CEO. And there's also a website where you can inquire more about corporate membership if that's of interest. I'm going to move swiftly on then to our program co-chairs. We've got Iona Agachi, IEC counsellor from Brasov in Romania, and Jiade Yu, an IEC counsellor from Boston, Massachusetts. So you're in good hands for the rest of the afternoon. And I'm going to wrap up now and wish you a very successful and educational and entertaining session. Thank you for your attention.
Okay, I hope that Dr. Irvin convinced you to join us and to support this very important initiative, I would say, in terms of education and outreach to make the best for our patients with atopic dermatitis. Now we start with the lectures, and the first lecturer is Dr. Zelma Ciesa Fuxenci from University of Pennsylvania. She's an associate of International Eczema Council, and she has a special interest in the diagnosis and management of adult patients with moderate to severe atopic dermatitis, heading efforts to develop a specialty program focusing on patients with complex disease at UPenn. She has received funding through the National Institute of Health, National Psoriasis Foundation, Women Dermatology Society, and Dermatology Foundation. And more recently, she is the recipient of a K23 Mentored Patient-Oriented Research Career Development Award to examine filagreen pathogenic variants in Hispanic Latino patients with AD. She has collaborated with the National Eczema Association and with the Asthma and Allergy Foundation of America, and is involved, as we already know, in multiple clinical trials for AD as principal investigator. She will talk us today of the role of peripartum antibiotics in the development of AD, a very interesting and timely topic. So please go ahead.
Hi, good afternoon, everyone. To my friends who are sitting on this side of the audience, I see you. I know you're there. It's a very big room. Thank you for the very kind introduction and for the invitation to be here today. And thank you to our audience for joining us this afternoon. So in the next 20 minutes or so, we're going to be discussing some of the most recent data around the role of peripartum antibiotics in the development of atopic dermatitis. I am a dermatologist by training, and I am part of a small group of dermatologists whose work also focused on dermatoepidemiology. So we're going to see a lot of tables and numbers, and I do apologize for that in advance. These are my disclosures of interest. And before we begin, I do want to take a moment to clarify a few terms that you may run into in the literature if you're interested in this topic. So the terms perinatal and peripartum are often used interchangeably, but they mean different things. For example, peripartum refers to the time from conception to first year postpartum, whereas the World Health Organization defines the perinatal period as the period from 22 weeks of gestational age to 7 days after birth. In addition to that, you are going to run into studies that use the terms antenatal, postnatal, perinatal, referring to 14 days after birth specifically, and intrapartum. And the reason why I bring this now is because if you look at the data and you're trying to make sense of it, I think that first and foremost, there needs to be consensus in future studies in terms of what definitions we are going to be using. And second, if you're looking at the data and you're trying to interpret the results, it's important to understand those definitions to make sure that your conclusions are valid. And so, as a dermatologist, I would be remiss if I don't put pictures on the screen of what atopic dermatitis looks like. So this is probably the only picture that you're going to see on my presentation. We know that atopic dermatitis is a highly prevalent disease. It is primarily considered a disease of childhood, and there is substantial variation in terms of its prevalence and incidence across the world. We also do know that it is quite prevalent in the adult population as well. And while we expect that most children would eventually outgrow their disease, there's increased literature to suggest that a lot of adults will continue to have persistent disease. Atopic dermatitis is also associated with high healthcare cost and also marked disease burden. And while the etiology of atopic dermatitis remains to be fully elucidated, I think it's important, and we all recognize that it's actually quite complex, and that it involves an interplay of genetics, environmental, lifestyle factors, disbalances in immune dysregulation, and also changes in the microbiota. And so when we talk about the microbiota of the skin, we know that skin and also gut microbiota are very important, right? When we talk about the microbiota, we're actually talking about that constellation of bacteria, viruses, and other organisms that live on our skin. And they are oftentimes referred to as that second genome because they can actually influence human health. A healthy gut and skin microbiota is important for protection against pathogens, and it has also been shown to be important in preserving that balance between what's known as protective and harmful inflammation. Now, in the case of certain disease states like atopic dermatitis, we know that there are certain imbalances in the composition of the microbiota. We know that there is decreased diversity and imbalanced expression of certain microbes, such as Staph aureus. We do know that lesions of patients with atopic dermatitis tend to have a higher prevalence of Staph aureus, and that correlates with disease severity. We also do know that in addition to Staph aureus, other pathogenic organisms like Cutibacterium can be expressed, which also increase the pathogenesis of Staph aureus. And then when we look at the gut microbiome in patients with atopic dermatitis, we know that this also can be quite different from healthy controls. Again, we decrease diversity of important bacteria such as Bacteroides, Lactobacillus, and Bifidobacterium, among others, and increased expression of pathogenic bacteria such as E. coli. And so while it remains to fully understand how is it that the skin microbiome and the gut microbiome come together to affect or improve or damage the skin barrier and how that it relates to atopic dermatitis, I think one thing is for sure, and we know that there are external factors that can affect the composition of the microbiota, one of them being exposure to antibiotics. And why are we interested in this work? And it's because if we can understand how antibiotics affect that composition, could we develop new theories or hypotheses around how they contribute potentially to the pathogenesis of atopic dermatitis? And again, why do this work? As I previously mentioned, atopic dermatitis is a highly prevalent disease associated with significant disease burden and high economic cost. We also know that antibiotics are probably one of the most commonly prescribed medications during pregnancy. It is estimated that worldwide, about a quarter of women or females who become pregnant will receive an antibiotic during their pregnancy. And it's also known to be very commonly prescribed during early childhood. Depending on the population studies that you look into, you will find that about 50% of children actually do get exposed to antibiotics before the age of 2. And in about a quarter of those cases, they get exposed to multiple courses. In addition to that, as I mentioned previously, if we can understand how antibiotics, if there's what is the baseline risk, could that lead to the development of other theories or hypotheses to explain that association? And does that have implication for other illnesses that share a similar nature to atopic dermatitis, meaning other atopic illnesses? And could it also help us develop novel preventive and therapeutic approaches? So what does the data show us so far? So in a recent systematic review and meta-analysis of all observational studies that were published between 2002 and 2022, the investigators set out to answer this question. So after going through the inclusion-exclusion process, they included 18 studies, both from Europe and Asia. They included a total of about 5.3 million mother-to-child pairs, so we're talking about a very large sample size. And they looked at antibiotic exposure across the board, either as recorded by records, patient questionnaires, etc. And their outcome of interest was the development of atopic dermatitis before the age of 18. These investigators found that looking at the large body of data so far, maternal exposure to antibiotics seems to be associated with about a 14% increased risk of developing atopic dermatitis in childhood. This is in contrast to a prior study that was published a couple of years before that showed a much higher risk, about close to 40%. Now, when they did subgroup analysis, and if you focus on the table that you'll see to your right, the findings did not seem to vary depending on the population that was being studied, whether it was the Asian or European populations. They did find that when they looked at certain types of studies, particularly retrospective studies only, that association was still maintained, but when they stratified and looked at other studies designed, that association was lost. And if I can just grab your attention and you can move towards the bottom of the table, when you look at timing of the exposure, what the investigators found was that if the antibiotics were administered right at the moment before delivery, it didn't seem to be affecting the risk. If it were administered solely during the third trimester, it also did not seem to be altering the risk. But if the antibiotic was prescribed throughout the entire pregnancy, it does seem to be associated with about a 10% increased risk in their offspring developing atopic dermatitis later on. So based on this review, what do we know? As I previously mentioned, there seems to be a biological plausible mechanism to explain this association. This particular study that I just mentioned included about 5.3 million mother-to-child pairs, so that's a large group of people, and their results were somewhat robust to sensitivity analysis. However, I caution you when looking at these types of studies because they have high heterogeneity. That means that it's very difficult to compare the data together, right? Because of differences in study design. There's differences in how the exposures were being measured. There's also differences in how the outcome was being captured. A lot of these studies did depend on the mother recalling that exposure, and that introduces bias, right? And it makes us question that association. So with that in mind, my mentor, Dr. David Margolis, posed the question to our research group, what is the true association of antibiotic exposure and the risk of atopic dermatitis? There we go. Okay. So our study, the initial part of a study was focused on looking at in utero exposure, right? So baby inside mom. And so we use something called the IQVIA Medical Research Database. So this is a database that's based in the United Kingdom. This is a database that's considered to be representative of the population. It covers about 10% of the population of the UK. And that data is available for researchers in a de-identified manner. The data gets captured in the database at the time that the patient goes to see their general practitioner. It gets recorded at that moment. In order for us to be able to match the mother and the child had to be registered within their general practitioner at least 60 days after birth. They have to have at least 2 follow-up diagnostic visits with the general practitioner. They had to use the same general practitioner as the mother and also reside in the same household. And in terms of antibiotic exposures, we measured this by the prescriptions that were recorded in the electronic medical record at the time of the encounter. And we categorized this into 4 different groups. Again, our outcome of interest here was incident atopic dermatitis in the child. As I previously mentioned, the other studies that have been published so far, there was variation in terms of the variables that were included. And we know that there are important variables that can confound this association, such as, for example, maternal history of atopic dermatitis, history of other atopic illnesses, Townsend Index, which is a measure of the socioeconomic deprivation of the person, ethnicity, and sex assigned at birth. And we followed these children also from the time of birth until they either transferred out of the practice, died, developed the outcome of interest, or it was the end of the data reporting period. In addition to that, we did separate sensitivity analysis where we realized that a mother could have more than one child. And we said, let's try to do a sibling cohort. Let's get a child from the same mother who has potentially the same genetic and environmental risk factors, and get one child that's been exposed and one child that hasn't been exposed, and we'll compare those two, because that allows us to control in part for those factors that may not be captured in the database. And so what did we observe? And I do apologize, this is very fine print, and I recognize this is a big room. But in summary, we identified about about 1 million mother-child pairs with data recorded between 2004 and 2021. Children were followed on average for about 10 years, and the mean age of the child at the time of AD diagnosis was about 3 years old. Oh, there we go. Apologize for that. Okay, now moving on to our analysis. If we look across the board, what this table here is showing you is the association between antibiotic exposure in utero and subsequent development of atopic dermatitis. And our findings demonstrate that children who are exposed to antibiotic in utero might be at an increased risk, about 38% increased risk of developing atopic dermatitis later in life. If you focus on the columns, they should be to your left, you will see that the risk seems to be similar despite the type of antibiotic that was being prescribed, with the highest risk seen particularly for penicillin. And it also didn't seem to vary depending on the trimester of the exposure, meaning if a mother was exposed during or took the antibiotic during the first trimester or the third trimester, the risk was still there. Interestingly, we did observe that this risk seemed to be higher in mothers in children of mothers who did not have a prior history of atopic dermatitis. So this is something that has not been previously published in the literature until our study, I believe, was done. In addition to that, what you're seeing here is the analysis of the sibling cohort, and you can see as well that our findings were robust to this analysis, again, with children who were exposed in utero to antibiotic having higher risk compared to their siblings who weren't, and that risk being highest in children of mothers who had no prior history of atopic dermatitis. Now, with any epidemiological study, there's bound to be limitations, right? And I think it's important to recognize those, right? We want to be fair and just. So there could be errors in how we lengthen mother and child pairs. It's important to note that we used a method that has been previously validated in the literature and used in similar types of studies. There could be information bias or ascertainment bias in terms of what data gets recorded in the medical record. However, we could limit that because the data was being recorded at the time of the encounter and did not depend on the mother recalling the exposure later on. There's also the issue of unmeasured confounders, but if we look at our sibling analysis, we were able to adjust for some of those things by doing that and showed the risk was not altered. And there could also be medications that were prescribed in the hospital setting that we couldn't capture because the nature of our database at the time didn't allow us to do that. In terms of the strengths of our article, is a large sample size. Prior to that, the systematic review and meta-analysis included about 5 million mother-child pairs. In our single study, it would include close to 1 million. And again, the data was collected at the time of the encounter. And in addition to that, because we know that antibiotic exposure, it's very hard to model, we created models that allow for that variation in the antibiotic exposure to change through time, providing for more robust analysis. So, what about then early life antibiotic exposure? So we're now moving to the child who has been born, who's getting that exposure at that very early moment in time. And again, a caveat here, when you look at the data of early life antibiotic exposure, there seems to be no consensus in the literature about what we mean by early life exposure. Some studies focus on 1 year, others mention 90 days, which is the case of our study, others 6 months, and others focus specifically on the perinatal period, which is that period from birth to 14 days. So, what did we know so far before we set out to do our study? Again, we revised the data and looked at some published studies. The most recent systematic review and meta-analysis that I could find on this particular topic was actually published more than 10 years ago. So we really do need to go back and look at the breadth of the work that's been published since then. And so this particular study included around 17 studies that evaluated postnatal exposure. Most of the studies were longitudinal, observational in nature. Again, there were ample differences in terms of how the exposures were being captured as well as the outcome of interest. But despite those limitations, what the researchers here found was that antibiotic exposure in the first year of life was associated with about a 40% increased risk for developing of eczema later in life. But we do have to take this again with a grain of salt just because these studies are all very different in how they're designed. They also observed that when they stratified or did analysis separately between cross-sectional studies which are basically just a snapshot in time, and cohort longitudinal studies where you do follow these patients into the future, they found that the risk was still the same. And so in addition to that, what I also found interesting about this study was that they also looked at studies that captured not only the exposure, but the courses of antibiotics. As I previously mentioned, there is data showing that about 25% of children may actually receive more than one course of antibiotics before they hit the age of 2. So it's important to understand how that variation also affects this risk. We don't have a lot of great studies in this area. We were going to focus primarily on the 2 studies that were used to actually show that exposure to multiple courses of antibiotics may actually increase that risk further by about 7%. And so in the study from Smith, which was published again a while back ago, while they did not find a difference between patients who were prescribed one course or patients who were prescribed no antibiotics, they did see about a 2-fold increased risk in the potential development of eczema in children who were prescribed more than 2 courses of antibiotics during the first year of life. And then the study by von Mutius, which is again, it's an older study dating back to 1999. So there's a lot of work that still needs to be done in this area. They also did observe that as you increase the number of antibiotic courses that a child is exposed to, that also seems to be having a dose response where the risk for developing atopic dermatitis later on increases. Now, what about more recent data? And this is a study that was published using a cohort from Sweden. And why am I showing this study here? And it's because this study, I think, does a fair job in addressing something we call protopathic bias, right, or ascertainment bias. So when you look at these types of studies, you might think oh, maybe this child who received an antibiotic early in life received the antibiotic for an early manifestation of atopic dermatitis, and therefore that's why you're seeing that association later in life. And so what they did was, instead of looking at the different antibiotic classes, such as penicillins, cephalosporins, etc., they looked at why that antibiotic was being prescribed in the first place. And so you could say, I see the risk there for patients who were prescribed the antibiotic for a skin infection, but kind of dubious about those results. But then when you look at patients who were prescribed antibiotics early in life, for example, for airway disease or urinary tract infections, you can see that that risk for atopic dermatitis does hold later on. So that tells you that there's probably something there, that relationship is true. And more recently, this was a study that focused on examining the association between antibiotic exposure very early in life, right? Some studies do it up to a year. This particular study focused on exposures during the first 6 months of life, and they did also additional analysis only focusing during that neonatal period. So this is a study that used the Southwest Finland birth cohort. At the time of the study analysis, it included close to about 11,000 children. 12% of them had been exposed to an antibiotic during the neonatal period, and about 16% of them did receive antibiotic exposure at some point after the neonatal period, but before 6 months of age. So that, again, supports the idea that antibiotics are actually very frequently prescribed early in life. In terms of follow-up, these patients were followed for about 8 years after birth. And what you're seeing here, if you look at figure 8, which should be to your left, you are seeing that their findings do support an association of an increased risk with exposure to antibiotics during the neonatal period, about 50% compared to neonates not exposed. And they also observed that on the figure to your right, that antibiotic therapy by 6 months of age was associated again with that about 38% increased risk of the child developing atopic dermatitis later on. But what about our study, right? As I previously mentioned, our study used a population-based database from the United Kingdom that is thought to be representative of that population. And here our children were followed for about 10 years, with the median age of atopic dermatitis diagnosis being around 3 years. And again, we identified about 1 million mother-to-child pairs. And what we observed, if you look at exposure to antibiotics early in life, you can see that towards the bottom of the table, about 72% of our cohort at some point had received an antibiotic, with antibiotic exposures again being more common in the patients who had a history of atopic dermatitis. When we look at the association between exposure to antibiotics and development of atopic dermatitis, if you look at the bottom line of table 3, you will have what is known as our adjusted model, and that includes things like maternal history of atopic dermatitis, maternal exposure to antibiotics, the sex of the child at sign of birth, and the child's other allergic illnesses, a measure of socioeconomic deprivation and ethnicity. And what we observed is that across the board, despite the type of antibiotic class that was being prescribed, children who received antibiotics had about a 40 to 80% increased risk of developing atopic dermatitis by the age of 3 when we adjust for those other confounders. And then we said, let's restrict our analysis to children who were only exposed during those first 90 days of life. And we also found that that association still holds true. So as I near the end of my presentation, what are some of the taking points that I want you to remember from this today? It's not that antibiotics are bad and that we shouldn't use them. That actually is not what we're trying to achieve here. I do want you to think about antibiotic exposures either in utero and during the early postnatal period as a potential risk factor for atopic dermatitis later in life. And why? Because contrary to perhaps other risk factors for atopic dermatitis like genetics, which are very hard to modify, this is a modifiable risk factor that we should look into. Our findings do not prove causation. So please don't go out and say, yes, she told me antibiotics cause atopic dermatitis, because that is not what we're doing in this types of analysis. But it does raise very important questions about future studies. And again, we do not advocate for not using antibiotics, but being judicious with their use, particularly in populations who might be at higher risk. And with that, I will come to the end of my presentation, and I would like to thank my primary mentor, Dr. Margolis, for his support, our research team at AD Club, which meets every Wednesday via Zoom Eastern Time, so you are welcome to join us if you're interested in discussing ideas, our co-authors on this paper, the Center for Clinical Science and Dermatology at Penn, and the Department of Dermatology for their support, and again, the International Eczema Council for the opportunity to be here and present our work. And I encourage you that if you're not a member, please come and join us, and thank you for your time.
We will have the discussion at the end with all the speakers. So the next speaker needs no introduction, but I will read it anyway. So it's Dr. Anne-Marie Singh from Wisconsin School of Medicine and Public Health. She's a professor of pediatrics and interim division chief of the Division of Allergy, Immunology, and Rheumatology, with affiliate appointments in the departments of dermatology and medical microbiology and immunology. She's also the director of the Food Allergy Research and Educational Clinical Research Center of Distinction, and as we know, she performs translational and clinical studies focused on pathogenesis of atopic dermatitis and atopic march. So she will tackle today a very, very interesting topic and bringing very new data from the environmental risk factors in atopic dermatitis, looking at the Children Respiratory Environmental Work Group Consortium and childhood allergies and the neonatal environment. So please, Anne-Marie, let me just try to put your... It stays a while with disclosures, but then it should not.
Okay, perfect. Thank you for that incredibly kind introduction. Hopefully, okay, here we are. Okay, so I'm going to talk about environmental risk factors in kind of 2 birth cohort studies that we have been working on. Okay, so for this audience, I don't need to say too much about this, but we know that, the atopic dermatitis prevalence has through the Isaac studies, it looked like it had been increasing, but then perhaps leveling off in our quote unquote westernized countries, but continues to be an important clinical issue in less developed nations. In industrialized countries, 20% is often what's quoted. And in Madison, Wisconsin, in our childhood origins of asthma birth cohort, it was 26%. And not only is atopic dermatitis common and potentially becoming more common, but as we just heard, it's a risk factor for the development of other allergic diseases, such as, of course, food allergy, asthma, and allergic rhinitis. And that's been termed kind of the atopic march, which you can see on the right-hand side of the slide. If you notice the references, when we talk about the epidemiology of AD, they are old, right? So it was sort of like once that final Isaac phase 3 was published, like maybe, especially in the US, like the book was kind of closed, right? Like maybe eczema had leveled off and that's kind of where we are. So I wanted to ask if that were actually true, so I wanted to update, like what does atopic dermatitis actually look like? In the United States. And I also wanted to think about the atopic march in a more nuanced way, because as we all know, especially as allergists, that patients they don't go through this really strict march of everybody with AD also gets food allergy and then gets asthma and then gets allergic rhinitis. And we know that there is a heterogeneous expression of that co-expression of atopic diseases. And so I wanted to understand that a little bit better. So to do that, I participated in this consortium called the CREW Consortium, which is funded through the NIH ECHO program, which stands for the Children's Respiratory Environmental Work Group. And here you can see we took data from 11 birth cohorts across the country that were focused on atopic dermatitis and asthma, but basically focused on allergic diseases. And I wanted to harmonize the data across all of these birth cohorts to understand what AD looked like throughout the United States. And so here you can see a map of all the birth cohorts that participated in the study, all the centers that participated in the study, and then the names of the cohorts and the number of participants on the slide. So my hypothesis for the study is that AD is a heterogeneous disease and that there are distinct AD expression patterns within CREW, and that these AD expression patterns would have distinct environmental risk factors and also a differential risk for other allergic disease development, right? So what are the expressions of AD? What are the environmental risk factors associated with each of those expression patterns of AD? And finally, depending on what your AD looks like, how does that predict your risk for the development of other allergic diseases? So to address this hypothesis, there are 3 aims that are listed on the slide, which I actually just talked about. Okay, so the methods. So we did a latent class analysis of the longitudinal expression of AD to determine AD phenotypes. And then we looked for associations of the other variables or risk factors that could be associated. So we looked at associations of other allergic diseases, again, to get a better nuanced view of the quote-unquote atopic march. We also wanted to look at markers of basically Th2 inflammation, so allergic sensitization and other Th2 markers like peripheral eosinophilia, total IgE, and then obviously other environmental exposures, and we looked at, of course, unadjusted and adjusted odds ratios. And then we constructed conditional inference trees to determine which factors are most likely to influence the phenotype. And so now you can kind of see what we looked at the bottom of the slide with our main diagnoses all the way on the left, and then our measures of allergic sensitization and like kind of Th2 skewing, of which I actually took that data out of the slide deck just to focus on the right-hand side, which are our environmental exposures. So to the point of our previous speaker, what time frame are we talking about with those environmental exposures? And we chose to focus on the first year of life. So we looked at birth mode, birth order, pet ownership, breastfeeding, respiratory virus infections, smoke exposure, siblings, daycare attendance, kind of our usual suspects for environmental exposures. And so because we are looking at the longitudinal expression of AD over time, we really wanted to include the cohorts in the study that assessed AD throughout childhood. So we wanted there to be AD at least 3 different time points. And so these are the cohorts that ended up being included in our analysis. You can see there's a mix of both population-based cohorts, which is about 2/3 of our cohorts, and 1/3 were high risk, meaning there was a parental history of atopy. You can see the cohorts span time. So we're talking from the 1980s to the 2010s. And then representative of the United States, you can see we do have about 60% of the cohort was white, about 20% was black, and the rest were other. Okay, so to answer the first question, what was our AD prevalence in our cohorts across the lifespan? So if you look at so first of all, to start off, AD was actually really common, and I was a little bit surprised by this. If you take each time point, the rates of AD were between 23 and 36% across these birth cohorts. And the next question we wanted to ask was, maybe that's because, as I mentioned, about 1/3 of those cohorts were high risk. So maybe it was because these families were high risk. And that actually is not what we find. We find that the rates are similar in the general and high risk cohorts. If you are born in a high-risk cohort, your risk of AD is higher, but only really early in life, until about age 2. And actually, then the opposite happens. The older you get, your AD risk goes up if you are in a population-based cohort. So it's interesting. And then here you can see the rates of AD at each time point in each individual cohort. So the graph on the left is just separating my high risk and general risk, and my graph on the right is illustrating the rates across each individual cohort. Okay, so now that, with the larger sample size, we're able to better understand that longitudinal expression of AD. And so earlier work that I had done as a fellow, actually in 2010, when we looked at just the Wisconsin population, we identified 3 expression patterns of atopic dermatitis. But now with this larger sample size across the US, we actually find, using our latent class analysis, the 5-class solution was our best fit. So here you can see our 5 classes of AD. In the upper left, the persistent AD. So these are kids who get AD early in life and it persists throughout childhood. The next you can see is the early AD with potential recurrence. So they have AD in infancy or until about age 2, and then it seems to improve. But then the longer you wait, it kind of rears its head again as the child gets older. There's a late onset class on the right, and then a group that has strictly early AD that does seem to outgrow it. And then finally our no or minimal or AD on the right. So next we wanted to look at, are there any predictors of which class, which expression pattern you would fall into? And after we adjust for cohort type, decade of birth, sex, and race, we find that children from high-risk cohorts are lower risk, as I mentioned, for the persistent and late onset AD, but higher risk for that recurrence. But moving on, kind of what we found was a kind of a striking relationship is that children of color are at higher risk for early expressions of AD. And when we look at, we find that Black children are actually at the highest risk for the early and persistent phenotypes. And not surprisingly, boys were also at higher risk for persistent AD. And so I apologize, I'm about to go through some tables, just like the last speaker. So they're all kind of formatted the same. So hopefully that'll help, where the phenotypes are across the top. And then if it's highlighted in red, that's a significant positive association, meaning that increases your risk. If the box is blue, then it's a protection. It's protecting. And then I try to summarize the main points in larger font next to each table. So hopefully that helps. So now if we want to look at the environmental exposures. So what are the environmental exposures that are associated with AD phenotype? We found that persistent AD was associated with actually breastfeeding, ever breastfeeding. So that's how we did that. And then antibiotic use in the first year of life. And this is actually in the child. So in contrast to the data we saw from the previous speaker, this is antibiotic use in the child in the first year of life. Daycare attendance also increased your risk for persistent AD, and then family history of asthma increased your risk. So that's kind of my pink boxes all the way on the right. In general, you can see those kind of all the little blue boxes kind of in the middle. Pet exposure in general was protective. So that's kind of what that is. And, we looked at whether it was in the first year of life or the first 5 years of life. We looked at dogs and cats independently as well. But in general, pet exposure was protective from AD. So next we wanted to look at how does your AD phenotype influence your timing, influence the development of the other allergic diseases. So this is to kind of answer that atopic march question. And here you can see each phenotype with the risk of allergic diseases listed on the slide. And after we adjust for some confounders, including cohort type, decade of birth, sex, and race, what we find is that early expressions of AD are associated, those are the phenotypes that increase your food allergy risk. The later expressions of AD, meaning late onset AD or persistent AD or AD with recurrence, those are the ones that are associated with increased risk for allergic rhinitis. And all of the phenotypes were associated with asthma, and we think this has to do with our asthma definition was pretty crude. It was ever asthma, like were you ever diagnosed with asthma? And we're currently looking at that in a little bit more detail. But if you have persistent AD, you are most likely to develop asthma. So this kind of begs the question, right? So the timing of AD suggests that's really important and what other allergic diseases you're going to develop and why might that be? And so when we think about the atopic march, I think, especially when we kind of look at the original postulations, there was a lot of questions or thought that maybe this is Th2 skewing, right? Like these patients are just more atopic, and so they get all the atopic diseases. So in our Wisconsin population, I was able to have access to basically serial blood samples starting from when the children were infants all the way until they were 18. And we were able to take those PBMCs, their peripheral blood mononuclear cells, and stimulate them and look to see, are they really like Th2 skewing, right? Is this really being driven by increased Th2? And interestingly, it didn't matter what time point we looked at, so you can see all the ages in the middle. It didn't matter what time point we looked at or what cytokine we looked at, there was no significant difference, right, between the phenotypes and their kind of Th2 skewing. So there's a couple explanations for this, right? So either I didn't look at the right way or I didn't do a detailed look enough to identify any systemic changes in the peripheral blood. Or another explanation is maybe all the action isn't happening in the peripheral blood, but it's happening somewhere else. And, obviously, we would start thinking about the skin barrier, right? Like what is happening at the skin barrier? So obviously a larger sample size will allow for a more nuanced explanation of this. And now in our CREW consortium, we are now going to start looking at, what are some factors in the epithelial barrier, including genetics and epigenetics and metabolomics to understand that. Okay, so why is AD more common now, and why is it at certain ages associated with specific allergic diseases? Again, as I mentioned, I think this suggests a role and a crucial role for the epithelial barrier. And so what are the mechanisms by which this is going to happen? So when we think about the epithelial barrier, right? Like this is where the point of first contact, right, of our immune system, whether it's pollution, the microbiome, viruses, or allergens, right? Like that is the point of first contact to these environmental exposures. And based on that content, that can influence both your epithelium, so it can definitely affect our barrier function, and as well as then kind of the what happens kind of either locally in the skin or systemically with Th2 inflammation. So we all know about our epithelial cytokines, TSLP, IL-33, IL-25, which can then, activate ILC2s and Th2 skewing and maybe promote some dysregulation. So I want to kind of zero in on one of our studies that's trying to understand that. And so I'm going to start with a study that we did of the microbiome. So this is a pilot study that we did. So it's looking at, it was just 20 children with AD, and we swabbed their skin. And so we have non-lesional and lesional skin. And from those skin swabs, then we were able to do detailed metagenomic sequencing to try to understand the structure and function of the microbial communities that are on the skin. We were then also able to, isolated individual isolates. So we could get down to the specific bacterium that was on the skin. And with those individual isolates, we can then take those in vitro and see how they may influence keratinocyte biology, as well as perform whole genome sequencing on those isolates to understand what they could be doing or to the keratinocytes. So here's some data on the right that's kind of illustrating, as we just heard, dysbiosis is a hallmark. I should have said that first. Dysbiosis is a hallmark of AD. And we always focus in on Staph aureus, I feel like, when we talk about AD. And so you can see my Staph aureus are my yellow dots at the bottom. So we definitely find Staph aureus, and we only find Staph aureus on our patients with AD and not on my control children. And then here, however, you can see we find many other staphylococcal species. And so I guess what I want to raise here is that I don't think it's just aureus that is promoting epithelial barrier function. I think there are other staphylococcal species and strains that promote barrier dysfunction. And it's not just, you have to go beyond species and down to strains. So if we look in the middle at Staph epi, these are my navy or my dark blue dots. And so here, if you look, let's see. Yeah, so the strain 1361, let's see. see, I can barely see it here. So here, if you look at strain 1361, we only isolate this strain of Staph epi from control children in non-lesional skin, and it's absent from my lesional swabs, suggesting perhaps this could be a protective strain of Staph epi. But here we have these other strains of Staph epi that we only isolate from patients who have AD, and some that we only isolate from lesional skin. So just kind of suggesting that it's very likely more complicated than just Staph aureus kind of being our only player. So what is it about these bacteria that can be promoting dysfunction? And so this is a heat map now. So if you remember, I said we isolated each individual isolates and did whole genome sequencing on the bacteria. And here you can see basically, then we looked at expression here of basically virulence factors, toxins, enterotoxin. And here you can see like some strains, like on the left, they're making a lot of bad things, right? Like there's a lot of navy there, so they're making a lot of virulence factors and toxins. And actually those strains are the ones that are associated for my patients who have AD plus food allergy, where the other strains here, which are have less of the virulence factors, are for my patients with AD alone. So again, suggesting that who is on the skin may also be influencing who goes on to develop other allergic diseases. The graph on the left is looking at focusing in on my staph, expression of the staph enterotoxins. And so here you can see strain 8. Oh, the strain isolated from patient 8, their non-lesional strain here, their staph makes a lot of enterotoxins, including SEB, SEC, and that patient had more severe AD. That's what the 3 hashes mean. It means they had severe AD as well as food allergy, where the other 2 participants had AD without food allergy. And so here you can see less toxin expression. So then we took these individual strains and we incubated them in a keratinocyte culture, and then we can measure the transepithelial resistance across that barrier. So this is now an in vitro measure of keratinocyte function, meaning are the keratinocytes functioning properly? And here you can see kind of exactly what I was saying. If we just highlight here my 3 Staph aureus strains that we isolated, those all significantly reduce my TER, my transepithelial resistance. And then we find other strains that we think are actually quite important, including some capitis and then hominis and some of our others. And then here we can see that variable effect depending on the strain of Staph epi, where these 2 strains here, they actually are protective. They actually significantly increase my barrier function. So they have higher TER, where kind of these other strains are lowering the TER. So again, suggesting that this environmental exposure of the microbiome on the skin is influencing, first of all, so it's influencing the severity of their AD, it's influencing who with AD develops food allergy, and potential mechanisms could be through the expression of these toxins and virulence factors that are affecting my keratinocyte function. We took the supernatant from these cultures and looked at cytokine expression. And so here's some data looking at TSLP. And so here you can see the staph isolates that were derived from non-lesional skin caused increased TSLP expression from the keratinocytes compared to the isolates coming from controls. And then here again, we can see a species level difference based on the species here. And we have further data looking down to the strain. But again, Staph aureus definitely caused increased TSLP, but so did capitis and Warneri. So thinking like, again, it's not just a single player. So how do we want to take this moving forward? So this takes us now to the CANOE study, which stands for Childhood Allergies in the Neonatal Environment. And this is our birth cohort to study a diverse population across ethnicities, lifestyles, and geographies. So you can imagine if I were to enroll another birth cohort in Madison, Wisconsin, maybe all my patients would look the same way or be kind of very similar. And if we really want to try to understand the role of the environment, I need other environments, right? Besides my upper Midwest college town. So we enrolled at 4 sites: St. Louis, Nashville, Madison, and Detroit. And these sites were chosen specifically because of their experience with birth cohorts, but also to get a mix of population density, so rural, urban, suburban, as well as ethnic, racial and ethnic representation. And this is funded through the ECHO program. So in addition to kind of my asthma and allergy outcomes, we are also collecting data on neurocognitive outcomes, obesity, and overall health through the ECHO program. So this is some preliminary data. So the children now are between 2 and 3 years of age. So if we look at AD prevalence at 12 months, and again, this is like really busy, but just kind of showing you like these are kind of on a first look, kind of the factors that we looked like. And then you can see which are the ones that increased my AD risk. And basically, similar to my crew data, race was actually the strongest predictor of who was going to get AD, with Black children being at highest risk. Maternal history of AD or food allergy also increased your risk, but interestingly, not other allergic diseases. So maternal asthma didn't increase your risk. It really had to be maternal food allergy or AD. And then, oh, thank you. And then environmental factors, season of birth was increased your risk. So those born in summer and fall were higher risk than those born in spring or winter. And C-section was associated with AD, and again, dog was protective. And so I mentioned we had done these conditional inference trees in crew, and when we look at this data, when we did those conditional inference trees in crew, the number one predictor of what phenotype you landed up in, like the first branch on the tree, was race. And meaning if you were Black or not Black was the first thing. And this is a strong thing, a strong predictor in canoe now too. So what we want to do is understand why that is, right? So I certainly do not believe that the skin of Black children is different than the skin of, any other person. And instead, I think in the field of medicine, race is often a proxy for other exposures that are harder to measure or that we don't always investigate very well. And this includes the role of the environment. And so we all know, hopefully this isn't surprising to anyone, right? Well, we all know that there are very important social determinants of health that kind of predict our disease expression. And when we think about in the world of allergy, air pollution, distance to traffic, housing conditions, neighborhood and built environments, economic stability, access to healthcare, all of those factors can increase stress, but also maybe have a direct effect, right, on these patients to increase their allergy risk. So to try to understand this, I want to untangle, right? So this is what this latest project and grant is about, is to try to untangle that risk of why is it that race is such a strong predictor. We are, in collaboration with Pat Ryan at the University of Cincinnati, we are taking the addresses of every canoe participant and where they live, as they move, and based on that address, we are geocoding that address to then get at all these kind of neighborhood level factors that you maybe can't get on a questionnaire. So that includes neighborhood factors, access to grocery stores, access to green space, pollution. So we're looking at nitric oxide, carbon monoxide, carbon dioxide, distance to highways, green space, childhood opportunity index, as well as the microbiome. And so then we can link their addresses to all of this environmental data. And so we are just starting that now. We have, Pat has an oral abstract later in this meeting. And one of our strongest predictors is distance to highway. So how close do you live to a highway? Predicting your AD risk. So with that, I'm going to end and just summarize that AD is very common, and all those epidemiologic studies that we talk about are old, and now with our newer look, it's about 25 to 30%. So I think it's probably still increasing. AD is heterogeneous with expression patterns that are variable throughout childhood. We have both demographic and environmental factors that are influencing AD expression, and you can see they're listed on the slide. I tried to do a deep dive into one of our environmental factors that we're looking at, which was the staphylococcal strains that are differentially associated with AD expression in the atopic march. And we think that the environmental effects on the epithelial barrier could provide a mechanistic link of how your environment shapes your allergic disease risk. So with that, I will just like to thank my lab and my collaborators who contributed to the work.
Thank you for the great lecture and also for the help that we had for organizing this symposium, this educational course. Okay, now we will, we will go ahead to, we will keep to the epithelial barrier theory with the next one. Okay, here we go. So I will focus mainly on the crosstalk between the skin and the lung. I will leave the gut for the next talk, mainly. These are my disclosures. None of these are relevant for the talk that I'm giving today. And again, we start with a nice picture of the atopic march. But just to make, again, this is not valid anymore. We know that there are different endotypes there, and it looks pretty much different in terms of curves that are occurring there. But one point that I want to make, it all starts in two sites that are very important for our thinking. One is the skin, so something happens there, and one is the gut. So very early events happening at two sites that talk to each other and also influence what is very distant, you know, the airways, the upper or lower airways at some time point in life. So probably you are aware with this concept, which is the epithelial barrier theory that was developed by Professor Ardis in Davos. And here, the multiple exposures that come through the lifetime, like for example, composing what we call the exposome, the allergens, the pollutants, the microbiome dysbiosis that we just heard about, the decreased biodiversity, they all create this epithelial damage. This epithelial damage and stimulation, which results in release of the epithelial-derived cytokines, stimulation of the immune response, and then the creation of inflammation in the tissue. But because the epithelial is leaky, then the microbes get into the tissue and they also contribute to this vicious circle that is occurring in chronic diseases. Furthermore, the disease doesn't stay only at the local site of injury, but gets systemic. So this circulating inflammation is very important because it's also a mean of communication between different epithelial barrier sites. This response, this epithelial barrier response to stress is a stereotype response that occurs at all barrier sites. Either it is driven by, for example, dietary antigens, additive and surfactants, or in the sinus by pollutants and allergens, or in the skin by different exposures. They all lead to the same type of inflammation and response. And with this in mind, we created a list, let's say, of what is a disease that is driven by this epithelial barrier defect. So it has to have increased prevalence in the latest, after 1960s, without being better diagnosed. For example, eosinophilic esophagitis or atopic derm is still the same way diagnosed, but we see a very steep increase in prevalence. We have to have proof of epithelial barrier defects and epithelitis, microbial dysbiosis, circulating inflammation. And usually this occurs, as I say, because it's a stereotype response of different size as multimorbidities. So where and all it starts? So now I will refer to the relation between the skin as a mirror for the lung. I'm mainly treating patients with asthma. So this is how it starts, and I will present you the data that we have, looking at how the skin as a barrier can inform us about the lung. And we know that since this very famous mice model, that spontaneous lung inflammation occurs if you have a skin barrier defect. It's not only a T2 inflammation, but it's also with increased cytokines of Th17 and T1 immune response. So a broad immune response occurs if you have a skin barrier defect. Now, as you know, you can that you can look at the skin barrier in different waves and flavors. For example, you can look at the transepithelial resistance or different types of clinical signs that you see there, or you can go more in depth and look at genomics, epigenomics, transcriptomics, proteomics, lipidomics. Better if you do all of them all together. What I will show you today is some transcriptomic data that we gathered in help of our research. What we use this tape stripped skin. We already know that this is very good for showing that it's sensitive enough to show modifications that occur in non-lesional skin, and this has been done many years ago by the group of Emma Goodman in patients with atopic dermatitis. Why the tape strips? Because the tape strips have a better profile, for example, than skin biopsies in looking at barrier and immune factors that are dysregulated when the skin barrier defect occurs. So with that in mind, we were during the pandemics, we couldn't do much in terms of research. So the skin was readily available. And we said, let's look at the tape strips in healthy skin of children with allergic asthma. This is the group that we have. So children with moderate allergic asthma and severe allergic asthma versus healthy controls. You can see here that they were quite very well matched in terms of age and sex and race and other parameters that are here. And we did tape strips and we looked if there is a difference in completely healthy skin of these children with allergic asthma versus healthy controls. And of course, not to our surprise, because we were expecting this, we saw that there is a significant difference between asthma and controls. You can see that there is a very nice separation here between the groups of controls and patients with moderate and severe asthma, and also moderate and severe asthma, they cluster differently. There were a number of 1,000 genes which are upregulated in moderate asthma and twice upregulated or downregulated in severe asthma versus healthy controls. Okay, so our first hypothesis was that the skin barrier is impaired in children with allergic asthma, and we saw a significant downregulation of cytoskeletal organization-related genes in severe asthma and an upregulation of epithelial hyperplasia markers both in moderate and in severe asthma. Furthermore, there is a significant attenuation of terminal differentiation and cell adhesion genes in moderate asthma, and in severe asthma of the terminal differentiation of the cornified envelope, of seal adhesion, of keratin, and lipid biosynthesis and metabolism in severe asthma. When we look in the GSVA analysis, we saw that there is, besides the epithelial barrier defect that we have seen between healthy controls, moderate and severe asthma, there is an increased T1 immune response in the skin. This was a little bit unexpected because, you know, these are children with allergic asthma. So something happens in their lung and a different immune profile you have in the skin. This is very interesting, and I will come back with more data on how this signature varies according to age. Now, the second question that we had, we had the skin barrier defect in asthma. This was, let's say, somehow expected, but can the skin inform us of what is happening in the lung? So we look if the skin transcriptome can show some pathogenic pathways that are relevant for patients with asthma. So we saw that the tape strips capture relevant genes relevant to asthma remodeling, like, for example, genes that are involved in the airway smooth muscle associated mTOR pathway. That means the mechanism of hypertrophy in the airway smooth muscle cell. the mechanosensory associated with signaling, the ERP pathway, the TGF-beta signaling pathway, the cell stress response, the mitochondrial structure and function on one side. These are upregulated both in moderate and severe asthma. And some differences in terms of lesions that occur in the lesional pathway. So, for example, in the moderate asthma, you have the upregulation in the necroptosis pathway, and in severe asthma of the ferroptosis-related pathways. This is something that is already new in the pathogenesis of asthma, but this is happening and is mirrored in the skin. And we also have a very nice reflection of the dysregulation of the extracellular milieu in the lung parenchyma with upregulation of TIMP3, of TGF-beta co-receptor, and of mediators of cellular processing, and downregulation of extracellular matrix regulatory genes. Also, in terms of processes that are generating this remodeling in the lung, the escape trips informed on dysregulation of oxidative phosphorylation and aging mechanisms that are occurring in the asthmatic lung. Fine. So this is in the skin. Then we said, okay, let's have a look and check if this is corresponding to what we have in previous bronchial biopsies that we have from these asthmatic children. And you can see that the tape strips capture this regulation of 8 out of 12 genes that can be found in the biopsies of children with asthma. And these genes are related to host antimicrobial defects, defense apoptosis, airway smooth muscle dysfunction, and fibroblast activity. So finally, using the pathway analysis, we looked at what kind of pathways are relevantly upregulated in these patients. And you can see that there is a lot of novel pathways that are very interesting for remodeling, like, for example, the taste receptors and the taste receptor system, or the phosphorylation and oxidative status. Finally, the final outcome of this should be of clinical consequence. So we looked if these transcriptomic biomarkers correlate with asthma-related outcomes and if they can discriminate asthmatic children from healthy controls. And they do, because they nicely relate with strong endpoints in a clinical evaluation of a child with asthma, like, for example, exacerbations, lung function, both in large airways, FEV1, or small airways, evaluated through impulse oscillometry, or in the terms of exhaled nitric oxide, so the T2 inflammation that is in the allergic lung. And the genes that are related are genes related to airway inflammation or hyperresponsiveness, RNA transcription-related genes, mucociliary activity-related genes, taste receptor, as I mentioned, as a new pathway, regulatory markers, and epithelial characterization markers. And to our, let's say, joy of this research was that the tape strip biomarkers accurately discriminated between allergic asthma and healthy controls with 100% if you use a 2-gene signature. If you have 1 gene signature, then you have also a very good discrimination between healthy and control. So a very easily to use biomarker to study the lung. I will come back to that at the end. Now, these genes that are here and they can discriminate, the first one is a tumor suppressor gene, which is linked to emphysema, so lung degradation. The other one is a negative regulator of catalytic activity of fibroblasts. This gene is a nasal cilia sensor for microbial glucose metabolism. This one is a regulator of fibroblast mechanoactivation. And the fourth one is a regulator of length and functioning of cilia. So again, very relevant biological targets that are explaining remodeling in the lung. As I was mentioning, I presented you the data for the children today, but this signature that we are seeing in the skin is age-specific, with different, for example, I was mentioning that in the children there was a very high profile of T1 immune response. It seems that this decreases over age. For example, in what we called in asthma the honeymoon period, when the symptoms reside, there is a decrease in all this immune response that is occurring in the skin, and then it comes back when asthma recurs. The only that is, let's say, a little bit stable is the Th22, which stays stable over the lifelong. I would like to invite you to the poster presentation of Daniel, which will be on Saturday, and he will discuss more in depth the age-specific signatures that we are seeing. So, in conclusion, the skin tapes capture a transcriptomic signature that reflects the asthma pathogenic pathway in terms of immune response, in terms of extracellular matrix dysregulation, smooth muscle hypertrophy, epithelial barrier defect, TGF-beta signaling, and oxidative stress. It's significantly correlated with asthma-relevant outcomes like exacerbations, lung function, both central and peripheral, and inflammation, exhaled nitric oxide, and as I shown, it distinguished with high accuracy the asthmatic children from healthy control. What could be the practical consequence of that, and what we are pursuing now, is that these tape strips, which we know are minimally invasive, they could be used to study the molecular mechanism for the asthma endotypes. They are obviously more easier to be done than doing bronchial biopsies or looking into the lung directly, and to monitor, especially to monitor changes in disease activity over time, being so easily to be performed serially. With that, I would like to close and, of course, thank my research team and the team of Mount Sinai that was instrumental in doing this research. And, of course, we are here to celebrate the 10th anniversary of the International Eczema Council. Thank you so much. Okay, and we go now to the gut, and it is my great pleasure to invite the next speaker, which is Dr. Sharon Chintrajak from Sean Parker Center for Allergy and Asthma Research at Stanford University. Her research is focused on developing novel therapeutic options for allergic diseases and unraveling molecular mechanisms driving therapeutic response. She's absolutely an expert in many food allergy, atopic dermatitis, and COVID-19 trials, and we have to thank her for having the FDA approval of omalizumab in food allergy. She's also a very good friend, so go ahead, Sharon.
Thank you so much for this invitation. I'm so excited to be here to celebrate the 10th anniversary of the IEC and to be speaking to you about bilateral relationships between food allergy and atopic dermatitis. These are my disclosures that are not really relevant to this talk today. I think we've seen variations of what the atopic march is. And we see that atopy can begin very early in life, and unless there is interruption, that you collect additional atopic allergic comorbidities like food allergy, asthma, and allergic rhinitis. But it's not just unidirectional, right? These other diseases influence the other atopic conditions as well. And we can see, certainly in the case of food allergy, how food allergy can exacerbate eczema and how asthma can be exacerbated by food allergies and vice versa as well. So many cohorts have shown that atopic dermatitis early in life influences the development of food allergy. The ALSPAC study showed that oozing and crusting rashes in infancy increases the odds by 5-fold for the development of peanut allergy. HealthNet showed us that the severity of atopic dermatitis can influence food allergy prevalence at 12 months of age. LEAP has showed us with each increase in SCORAD level, there is an increase in the number of food sensitizations. And lastly, as some of our speakers have shown us today, that the duration of atopic disease and atopic dermatitis can increase the prevalence and severity of disease, and that's true for food allergy as well. Dr. Lack proposed a dual allergen exposure hypothesis in 2008, and where cutaneous exposures to foods, especially when there's a defective epithelial barrier that's present, and in the absence of oral exposure of foods, can be sensitizing to foods. And I see Kari Nadeau in the audience here. She has this lovely little poem, "Through the skin, allergies begin, and through the diet, allergies can stay quiet." And the timing of that is so important. And we've learned that, in relation to the LEAP study in 2015, where early oral introduction of foods during infancy is essential for reducing peanut allergy at the age of 5 years. So the immune response behind atopic dermatitis and food allergy involves mechanisms that are complex and shared. There's genetic components, environmental components that lead to the impaired barrier dysfunction, and that contribute to mechanisms in the skin and the gut for IgE-mediated food allergy and eosinophilic esophagitis as well. Taking a closer look at this barrier dysfunction and its role in AD and food allergy, this is a nice concept by Dr. Bruff and others showing the outside-in and inside-out hypotheses. So skin barrier defects mediated by filigran deficiency, allergens, the environment, Staph aureus, as well as other staph species. We need to add a little asterisk there, contribute to a defective epithelial barrier and influence allergic inflammation on the inside. The allergic inflammation from the inside can disrupt the epithelial barrier, and then you're in this cycle that feeds each other to promote allergic disease. Many of our colleagues have shown that environmental exposures to food allergens can increase the sensitization and development of food allergy. And even consuming foods in the household, which is normal, can be found to have those proteins, those food proteins in household dust. And our colleagues have shown that, foods consumed in the kitchen, those proteins can be found in dust in the bedrooms, which are, sometimes separated by many feet and even different levels in a house. And for each unit increase in peanut dust concentration during infancy, that can have a 6-fold impact on food, peanut sensitization, and peanut allergy later in life. What we also don't know are what are the new factors from the environment and in a changing climate that contribute to a defective epithelial barrier, increased sensitizations. So transepidermal water loss tool is one of the tools in our toolbox, and really essentially measuring water loss from skin. So if you have a defective barrier, you have increased water evaporation, and that is captured by the tool measurements. And increased tool measurements have been shown in patients who have a filigran loss of function mutation at 3 months, even in the absence of atopic dermatitis. And tool measurements very early in life, 2 days of age and 2 months of age, have shown to predict atopic dermatitis at 12 months of age and predict food allergy at 2 years of age. So again, this sense that atopy is beginning very early in life, within days, and that if we don't stop this march, then allergic progression develops. Colleagues are using skin tape strips. We heard about this from Iona, looking for markers because this is a relatively simple test, minimally invasive, compared to lung biopsies or GI biopsies, where there are skin tape stripping measures to measure elements of the stratum corneum. And Donna Leung and colleagues looked at patients with atopic dermatitis and food allergy compared to patients with atopic dermatitis without food allergy, and did a multi-omics approach to look at what elements are different between these two patient populations. One patient population that has one atopic disorder, just atopic dermatitis, versus two, atopic dermatitis and food allergy. Lipidomics revealed that there's reduction of ultra long chain ceramides that are required for a normal skin barrier function. Transcriptomic studies showed that patients who have atopic dermatitis and food allergy have the highest type 2 immune expression levels. And proteomics revealed an immature keratin profile consistent with keratinocyte hyperproliferation in our dual atopic disorder and food allergy participants. Further studies looked at skin tape strips lipidomics, and this is a study published by Dr. Berdyshev in 2024, looking at a cohort of infants. And from that cohort of infants, 7% actually had developed food allergy alone, 7% developed food allergy and atopic dermatitis. And these are infants who had had skin tape strips done at 2 months of age and then looked to see about their food allergy status at 2 years of age. And these lipidomics from these skin tape strips profiles at 2-month-old babies. You know, we're not doing blood, we're doing skin tape strips, which is a relatively easy procedure in a 2-month-old baby. Even if they wiggle, you're able to do this. Can identify signatures that are food allergy specific with IL-33 and unsaturated ceramides, food allergy and atopic dermatitis specific, again with unsaturated ceramides, and then atopic dermatitis specific with a higher signature of TSLP. So how do we mitigate this effect in the development of food allergy? How do we try and treat atopic dermatitis to minimize the development of food allergy? There's been a number of different cohorts that have tried to look at this. And then I've summarized some of the main findings here. The BEEP study looked at petroleum or petrolatum-based moisturizers, and they looked at atopic dermatitis and food allergy and saw no reduction at 2 years of age of atopic dermatitis and a non-significant increase in food allergy in the intervention arm. The Prevent et al. Also looked at petrolatum-based moisturizers and did not see a reduction in AD at 12 months of age and no reduction of food allergies at 12 months of age. The Pebble study out of Australia looked at ceramide-containing trilipid creams, and there was a trend towards less atopic dermatitis at 12 months of age. And in the group that had excellent adherence, there was a significant reduction in food allergy sensitizations at 12 months of age. The PASSY study looked at proactive topical steroids, and in the group that used proactive topical steroids in less than 4 months of age, there was a 2-fold reduction of food allergy by 24 months. Our learnings from these different cohorts suggest that the earlier we are treating atopic dermatitis with not just improving skin hydration with petrolatum or creams or moisturizers, but also with ceramides and steroids, then we have a bigger effect on decreasing food allergy sensitization. So this is a very important area of investigation in the SEAL study. This is led by the NIH and Kari and Tina Cinder at Stanford and Kari at Harvard. Also Donna Leung at National Jewish, Chrissy Chow at University of Chicago, Gideon and Helen Bruff at King's College, and Neeru Hershey at Cincinnati's. Children's, and this is a 2-year phase 2 clinical trial looking at infants with dry skin or eczema. And the goal is to test whether aggressively treating dry skin and atopic dermatitis in early infancy can actually reduce the cumulative number of food allergies at 2 years of age. Again, infants are enrolled between 0 to 12 weeks of age, and we're looking at the number of food sensitizations and severity of eczema in patients who are receiving either ceramides and steroids or reactive care. And this study is currently still ongoing. I'm going to switch gears and talk about biologics. And I think I don't have to describe Dupixent, but it's an anti-IL-4 receptor alpha antibody, dupilumab, and it really inhibits IL-4 and IL-13 signaling, has downstream effects on B cells and class switching and IgE production and eosinophil recruitment, and is now approved for many indications, atopic dermatitis, eczema, EoE, nasal polyposis with chronic rhinosinusitis. And the very interesting part is that it's approved for children as young as 6 months of age with atopic dermatitis. There have been very recent studies showing that dupilumab alters protein markers that are associated with atopic dermatitis, and this is a study by Dr. Goliva, published just in January of this year, and looking at 20 patients with atopic dermatitis and 20 healthy controls who've received dupilumab. They looked at skin tape strips and did a heat map, which identified 2 clusters of proteins. So you have cluster 1 shown here and cluster 2 shown here. And in cluster 1, you can see in the lesional, the skin tape strips from the lesional atopic dermatitis regions show an increase in the cluster 1 proteins from the lesional skin and a decrease in cluster 2 proteins with dupilumab, and approaching the protein profile of those patients with healthy skin or non-lesional areas. We've seen now numerous studies of patients who have received dupilumab for atopic dermatitis on allergen-specific IgE, and particularly food-specific IgEs. So I'm showing you multiple panels here. This is from one manuscript, this is from another. But essentially, you can see that over time with dupilumab, we see a reduction in peanut-specific IgE, hazelnut-specific IgE, and we see various allergens here too: cashew, walnut, hen's egg, kiwi, apple, cow's milk, pistachio, almond, for not only the whole specific IgE, but the components as well, which is really exciting because we know that comorbid disease exists. And recently, the dupilumab food allergy study was published, and this was, Tina Cinder was the first author here, and we see the same allergen-specific IgE decreasing. Dupilumab was given to peanut allergic children, 24 children over 24 weeks, and then food challenges were done. Only 8% passed a food challenge to 444 milligrams of peanut. And this was a little disappointing, I think, for all of us in the food allergy community, especially as we see that the peanut-specific IgE is decreasing. So it's not the whole story, and I think that 24 weeks may be an early look, especially if we look back and see that we're looking over, 156 weeks. Maybe we just need a longer amount of time to see more than a 90% reduction in allergen-specific IgE before we see that as a clinical phenotype that might help our food allergic patients. The other dupilumab food allergy study looked at dupilumab as an adjuvant to peanut oral immunotherapy. This was a phase 2 multi-center study looking at 148 peanut allergic children aged 6 to 17. They were randomized 2-to-1 to receive dupilumab plus peanut oral immunotherapy or placebo plus peanut oral immunotherapy during a 28 to 40 week updosing period. And here we see that the addition of dupilumab did make a difference in the ability of participants to pass a double-blind placebo-controlled food challenge to 2 grams of peanut protein after 28 weeks of oral immunotherapy. So 56% of those in the combination arm, the dupilumab plus peanut oral immunotherapy arm, were able to pass that 2-gram threshold compared to 36% with peanut oral immunotherapy alone. So there's an additional benefit of using that combination approach. And interestingly, what I find very interesting and important is that the dupilumab was able to reduce some of the side effects during that build-up phase of peanut oral immunotherapy. For those of you who know oral immunotherapy, the main side effect and the most common side effect is GI intolerance. And this can actually be very burdensome to patients and their families and limit compliance and long-term feasibility with oral immunotherapy. So to minimize that side effect, I think, is one of the things that we're really hoping to do for allergen-specific immunotherapy. So in the dupilumab arm, there was a reduced proportion of days of GI symptoms related to oral immunotherapy, 5.1 compared to 17.8 in the non-dupilumab arm. And also notable was that there was no difference in the skin symptoms between the groups. We do know that oral immunotherapy can exacerbate underlying eczema. There's actually not a great recording of this. In my own experience, I see that it happens, but not in all patients that have atopic dermatitis. So this is fascinating to understand that particular phenotype. And I think the studies looking at patients with comorbid atopic dermatitis and food allergy that is current versus historical is really important to try and understand some of those mechanisms underlying that. We have a study called the Combined study that's actually combining 2 biologics. So starting with omalizumab to reduce circulating IgE, followed by dupilumab with multi-allergen oral immunotherapy. And this is a study that is currently still ongoing. And we're very curious to understand if the combination of biologics is effective in inducing sustained unresponsiveness compared to omalizumab alone, followed by multi-allergen oral immunotherapy, or dupilumab with multi-allergen oral immunotherapy. So again, the idea trying to see how quickly and effectively can we drive down allergen-specific IgE, and does that make a difference in the clinical phenotype for our food allergy patients? I'll switch gears and talk a little bit about JAK inhibition in atopic dermatitis and food allergy. We know that JAK-STAT signaling controls transduction through many inflammatory cytokines. JAK1 mediates signal transduction from key Th2 cytokines. And a meta-analysis suggests that JAK inhibition is an effective therapy for atopic dermatitis, looking at not only JAK inhibitors, I'm not going to say any of these names correctly, upadacitinib, abrocitinib, baricitinib, and in addition to dupilumab, we see that these are effective therapies for atopic dermatitis. And I was very excited to see JAK inhibitors applied in food allergy as well. And this was published in 2023, looking at JAK inhibition and T-cell and basophil and T-cell reactivity to peanut in vitro. And we see that there is a decrease in basophil activation and in a abrogated response of memory CD4 positive T cells upon peanut stimulation. So I think there are ongoing studies to further look at this in our food allergy patients, and we'll be excited to see what that looks like. And I will conclude that we know that there are many effective atopic dermatitis therapies out there, and we are very excited to see how they play out in food allergy. We know that AD is an important risk factor for the development of food allergy, and they share very similar molecular mechanisms, but there are some distinct mechanisms that are being identified through omic approaches and through minimally invasive testing, which I hope can be not only helpful in predicting which patients can go on to develop multiple atopic disorders, but also inform us on who would benefit from which therapeutics. And I think a very important question is if we can treat early our atopic dermatitis patients, can we prevent the progression of the atopic march? I didn't have time to talk about the Outmatch study, but it is now showing that omalizumab is now approved for children as young as 1 year of age and for food allergies. And as we look at these biologics and try and understand which patients we would want to use biologics, particularly in such a young age group, I think that's where there's the potential for immune modulation, and there should be very careful investigation there to see if we can actually halt the atopic march. So I'd like to thank my team here at the Sean and Parker Center and our funding sources, and again, thank the International Eczema Council for inviting me for this talk.
Thank you, Sharon. And we will have a 15-minute break. So I would like to ask you to be back at exactly 2 o'clock so that we can resume part 2. Thank you. We go ahead discussing the increased prevalence of allergic contact dermatitis in atopics, and it's my pleasure to invite the next speaker, which is Dr. Amber Atwater, which is the founding owner and manager of Distincting Dermatology in Vienna, and she is a clinical associate professor of dermatology at George Washington University in Washington, D.C. She's a national recognized expert in allergic contact dermatitis and patch testing, but she's also an expert in atopic dermatitis with extensive clinical experience in taking care of patients with this serious condition. She's a new associate of our International Eczema Council and past president of the American Contact Dermatitis Society. With no further ado, please, and thank you for stepping in and helping us with the symposium.
I feel like I need to disclose that I'm a dermatologist. There are a few of us speaking this afternoon, and I'm excited that to be here and speak with you today. My name's Amber Atwater, and we'll be talking about allergic contact dermatitis in patients with atopic dermatitis. So today we'll talk about the historical context and current understanding of allergic contact dermatitis in patients with atopic dermatitis. We'll think about some patch testing considerations in these patients, and we'll talk about some patch test results expectations in patients with atopic dermatitis who are undergoing patch testing. So what's the historical context here? The question that seems to be ongoing, and I think is still ongoing to this day, is can allergic or should allergic contact dermatitis and atopic dermatitis occur at the same time in the same patient? Going all the way back to 1964, a group from Marshfield, Wisconsin looked at 3 groups of patients, some with acne, some with psoriasis, and some with atopic dermatitis. And they looked retrospectively at patch testing for these patients, and you can see a very limited number of allergens were tested. What they found, and they looked at these 3 groups because all 3 of these groups would have been patients who had products applied to their skin regularly for their disease state. What they found was that none of the atopic patients had any positive patch test reactions. About 28 out of 100 atopic patients had positive patch test reactions, and you can see here that only the atopic patients had reactions to at least 3 substances. And then 9 out of the 100 psoriasis patients had positive patch test reactions. And the argument that this group made at this time was that these patients applied products regularly, but the atopic patients happened to also have an impaired barrier. And this was why they had an increased frequency of positive patch test reactions. This discussion is ongoing, but more recently, in 1973, which is many years ago at this point, another group looked at military members and their dependents, and they looked at atopic patients and controls. This is more of a prospective study. And they asked these patients, okay, do you have a history of poison ivy, poison oak, or poison sumac? And a very low percentage of patients with history of atopic dermatitis said yes, versus a higher percentage at 37% of the controls said yes. And then they moved forward and they patch tested these patients with Rhus oleoresin, and those patients with no history of atopic dermatitis had a higher percentage of positive patch test reaction to Rhus, and those with history lower at only 15%. Now, we can argue that this is a pretest probability problem. If only 1 out of 40 have history of reaction to poison ivy, we're going to probably see a low percentage of patients with a high percentage of positive patch test reaction. But regardless, the conclusion here was that patients with atopic dermatitis were less likely to exhibit allergic contact dermatitis. And this pulls forward to a textbook, a pediatric dermatology textbook. This is the second edition of Hurwitz from the early to mid 1990s, where on the atopic dermatitis chapter, we see them saying that in fact patients with atopic dermatitis have a lower risk of allergic contact dermatitis, and they quote this exact paper. So this one paper with 84 patients is pulling forward to saying, we don't have increased risk of allergic contact dermatitis in patients with atopic dermatitis. So where does this come from? As you know, the classic theory here is that atopic dermatitis is more Th2 skewed versus allergic contact dermatitis, which is more Th1 skewed. And if you think about this like a seesaw, if we have more Th2, we should have less Th1. And so in theory, these two should not coexist. In reality, the 2025 version of this is that it's not so straightforward, right? We have many more factors involved with both atopic dermatitis versus acute versus chronic atopic dermatitis, and more factors involved for allergic contact dermatitis, bringing into question, can these two coexist? So asking the question again, can patients with atopic dermatitis develop concurrent allergic contact dermatitis? And I think we all know that in some cases the answer is yes, but let's look at the data. This is a meta-analysis from 2017 where a group looked at the association between atopic dermatitis and allergic contact dermatitis. And they looked at 417 studies, and they found 74 papers that met their inclusion criteria. So of those 74 papers, when they take the total population, they find that there's no correlation between atopic dermatitis and contact sensitization. However, when they look at a breakdown of smaller parts of the same study, they look at the general population, so papers that looked at general population studies. They found 5 papers that did such. And at the very bottom here, you see when they combine the data that there's an increased odds of allergic contact dermatitis in their patients with atopic dermatitis. On the other side, when they look at patients in referral populations or specialty patch test clinics, you can see at the very bottom here that when they combine all the data, you can see that your overall odds ratio is below 1 for atopic dermatitis and allergic contact dermatitis. So in specialty clinics, there may be an inverse association, so less allergic contact dermatitis in atopic patients. So why might patients with atopic dermatitis have a decreased risk of allergic contact dermatitis? We've already alluded to this a little bit. We've got this concept of the Th2 skewed inflammatory response. They should not coexist at the same time. And in that same vein, we can see increased elicitation thresholds in patients with atopic dermatitis as compared to those with controls. And for example, this is a study from 1989 that looked at 3 groups of patients with atopic dermatitis, those with mild, moderate, and severe atopic dermatitis. And what they did is they did what's called a dinitrochlorobenzene challenge. This is an allergen that's more commonly used in labs, not commonly used in clinics. But they challenged these patients with mild, moderate, and severe disease, and they found that 100% of patients with mild atopic dermatitis were able to elicit a response versus a much lower percentage of patients with moderate and severe disease. But interestingly, when they treated those patients with moderate and severe atopic dermatitis down to more mild disease, they were then, in many cases, able to elicit a response. So they had this increased elicitation threshold. Why could patients with atopic dermatitis have an increased risk of allergic contact dermatitis? Well, we know that patients with atopic dermatitis have a skin barrier dysfunction with associated increased absorption of allergens and irritants. We know that these patients are applying lots of things to their skin, whether they be over-the-counter products or prescription products. We know about these potential shared immune pathways that we talked about earlier. And we know that bacterial colonization may lead to enhanced sensitization and associated increased risk of contact allergy. So this is my very rudimentary drawing of the skin barrier. And let's think about how this works in the setting of this concept of allergic contact dermatitis. So in patients with atopic dermatitis, we can have the Lagran gene mutations, either endogenous or acquired. And we can see direct insults or epigenetic gene mutations from things like fragrances, pteritogens, stress, climate change, and pollution. All of these can impair the barrier. With this impaired barrier, we have an increased risk of allergen penetrance and an increased risk of sensitization and elicitation. So let's talk about patch testing considerations in our patients with atopic dermatitis. We're going to go through a few cases. Case number 1. This is a 21-year-old man with lifelong atopic dermatitis. His disease is always present in the anicubal and popliteal fossa, but for the last 6 months, he's had hand involvement. And so here you see pigmented papules and plaques with scale on the dorsal digits, and this involves both hands in this case. And so the question is, should you patch test this patient? If you're asking me, the answer is always going to be yes, but I'm going to explain to you why for each of the cases, all right? So this patient has new involvement on the hands. New involvement is a potential indicator for patch testing in atopic patients, as is involvement in a higher risk for allergy location like the hands. So other indications include atopic dermatitis that comes up in an adolescent or an adult, change in distribution or an atypical distribution of disease, a distribution that is consistent with allergic contact dermatitis like eyelids, periocular, periorbital, hands, feet, a change in intensity of lesions, a change in frequency or intensity of flares, and a limited change in or no response to therapy. The next case is a 63-year-old woman with a 30-year history of atopic dermatitis, and she's had worsening over the last 5 years, and she happens to be on a biologic when she comes to your clinic. Should you patch test this patient? She's had a worsening of her disease over the last 5 years, but then the question comes, she's on a biologic, what do you do? This is a paper just published this month. It is an update from the North American Contact Dermatitis Group on guidelines regarding patch testing on patients taking immunosuppressive agents. And there are 2 tables that I'm going to share with you. The first is the recommended hold time prior to patch testing for different medications. Now, I want to make clear here that this is consensus, and not every member of the North American Contact Dermatitis Group practices in this way. For example, I'm a member, I don't do this exactly how this is listed here or on the next slide, but I do it in similar fashion, okay? So for this patient who's on a biologic, the consensus is to hold the biologic for 14 days. There are some people who do this for 0 days, and there are some people who do this for 6 weeks. There's a group in Minnesota who holds for 6 weeks before they patch test. What I do is, if I can, I hold the week of patch testing or the week prior to patch testing. The next slide shows maximum recommended dose during patch testing. And what you see here for the biologic specifically, if it can't be held, you can still perform patch testing, keeping in mind that biologics may selectively impact patch test results for some allergens. And the ones that come to mind in the literature are mostly fragrances and metals. The other important point here on this slide is systemic corticosteroids. The general consensus for the group is that patients should be on 10 milligrams or less during patch testing. So this is a patient of mine. No photos of this slide, please. This is a patient of mine with atopic dermatitis who I saw just last week. And he came to me on mycophenolate mofetil 1,000 milligrams twice a day. and prednisone taper that I put him on just so we could patch test 10 milligrams a day because he was not clear on mycophenolate. And what you can see here is multiple positive patch test reactions in this individual despite being on these meds. Many patch testers believe that as long as the patient still has some dermatitis that you think is from the allergen at the time of patch testing, even if they're on an immunosuppressive agent, they should still be able to mount a response to the allergen. And in this case, in fact, we did. Now, you might ask, well, there's multiple positives, and that's just because he's an atopic and there's something wrong with your patch testing. The fact is, this patient had positives to probably 7 fragrances, 1 preservative, and a couple of surfactants, which is pretty classic for atopic patients and is why he has so many positives. The next case is a 37-year-old woman with lifelong atopic dermatitis with new facial involvement for about 4 months. And she uses hypoallergenic personal care products. And so what she's going to say to you is, I use hypoallergenic products. There's no way I could have an allergy. And in fact, I heard that yesterday in clinic, right? So the point here with this one is, should you patch test her? Of course you should. She has new dermatitis of her face of short duration. And this is my closet. I don't even have atopic dermatitis, but just imagine the patient with atopic dermatitis and how many products they might be applying to their skin, especially after their dermatitis worsens. And on top of that, they are also using multiple topical prescriptions. And we need to consider not only the active ingredient, but also the inactive ingredient for these patients. This is a study from 2017 that looked at allergens and popular moisturizers. They looked at 3 websites, popular companies like CVS and things like this. They looked for words like hypoallergenic, dermatologist recommended, fragrance free. And what they found is that out of 174 products that they found online, only 12% were free of North American Contact Dermatitis Group allergens. The North American Contact Dermatitis Group regularly tests about 80 allergens at a time. So there's 80 common allergens that were all over these products. And then when they broke down the allergens by product, they found that the most common allergen in these hypoallergenic products was fragrance, which is a very common allergen, paraben mix, and tocopherol. Now, I would argue paraben and tocopherol allergies are not super common, but if you go down to number 5 here on this list, formaldehyde releasers, common preservatives found in personal care products. So what do we see for patch test results in patients with atopic dermatitis? This is another study from the North American Contact Dermatitis Group where they looked at data from 2001 to 2016. And what I'm showing you here is patients with history of atopic dermatitis versus patients without. And odds ratio increased means that patients with atopic dermatitis in this case had a higher frequency of a positive patch test to the allergens that you see here. So the first 4 allergens here are preservatives. So we've got formaldehyde in 2 different concentrations, quaternium-15, which is a formaldehyde releaser, and methylchloroisothiazolone, methylisothiazolone, common in this group. 2 fragrances, fragrance mix 1 and colophony, followed by bacitracin, which is a topical antibiotic. This is where we get into the medicament problem for patients with atopic dermatitis. They're going to have an increased frequency of allergen to medicaments in general. So we've got bacitracin. Lanolin is a classic for patients with atopic dermatitis because this can be commonly seen in patient products. Oleimidopropyl dimethylamine, which is a surfactant. We'll talk a little bit more about that later. Carbamix and potassium dichromate, which is a metal. Another study from the United Kingdom looking at 30 years of data showed very similar results. Fragrances at the top, followed by neomycin, tixocordyl, which is a corticosteroid, lanolin again, and imidazolidinylurea, which is a formaldehyde-releasing preservative. What about kids? Is there increased risk of allergy in allergic contact dermatitis in kids with atopic dermatitis? Of course there is. This is data from the Pediatric Contact Dermatitis Registry based in the United States. And what they found was that there were 4 allergens with increased relevance in their patients with atopic dermatitis. And these were cocamidopropylbetaine, which is a surfactant, Will alcohol, aka lanolin, tixocordyl, a corticosteroid, and parthenolide, which is a plant-based product that can be found in some natural products. And as you know, atopic patients love to use natural products. And then another study in children showing that kids with atopics may be more likely to have more than 1 positive patch test reaction. and a greater number of positive results. And then you see the top allergens here with increased prevalence in patients with atopic dermatitis, where cocamidopropylbetaine, a surfactant, carbamix, a rubber accelerator, and bacitracin. So let's look at two more groups. The first is preservatives. Now, a group, Don Belcito, who was both in Kansas and New York City at one time, looked at his data for all of his patients patch tested over a time period, about 2,400 people. And this first figure that you see is just his overall population. Black is atopic patients, white is non-atopic patients. And you see that there's significance here for higher frequency of just positive patch test reactions in his atopic patients. When he focused in on preservatives, however, we see that there's significance for 4 different formaldehyde-releasing preservatives, which you can see here, but a numerical significance for standard formaldehyde and diazolidinylurea, another formaldehyde-releasing preservative as well. And then surfactants. The same group looked at surfactants. Surfactants are important in atopics because surfactants are hydrophilic, meaning water-loving. The skin, when it's normal, does not allow water to go through it, but when the barrier is impaired, it can. So products like surfactants have an easier way of affecting patients with atopic dermatitis than others. They're more likely to be an allergen in an atopic patient. So what you see here is atopics had a higher frequency of reactions to cocamidopropylbetaine and a numerical increase for amidoamine and cocamide DEA, 2 other surfactants. So what are our most common allergen in atopic patients? Well, fragrances and preservatives, sure, we see that in everybody, right? But we can see topical medicaments, including corticosteroids, antibiotics, and chlorhexidine. Surfactants, we've mentioned those already, cocamidopropylbetaine and dimethylamminopropylamine. Lanolin, because it can be found in medicaments, once again. I would argue that metals is more of a mixed picture. We see some increased and some decreased, and rubber accelerators. So with that, what are the clinical messages today? So first, the atopic dermatitis, allergic contact dermatitis interplay is complex, and I think the story is ongoing. Contact allergies should be considered in patients with atopic dermatitis and atypical clinical presentations. Common allergens include fragrance, preservatives, lanolin, medicament-related chemicals, surfactants, and I said medicaments already, and preservatives. And medicaments and emollients are frequent sensitizers and should be included in routine patch testing. And that means you're not only testing the individual ingredients, but also the patient's personal care products as well. Thank you very much.
Thank you. We continue with the irritant contact dermatitis in atopics. My pleasure to introduce Dr. Aquino from Brown University of Medicine School. She is, besides being professor of pediatrics at the Warren Alpert Medical School at Brown University, program director for Allergy Immunology Fellowship at Brown, Rhode Island, and Hansbrough Children's Hospital. But she's also the chair of the American AAAAI Allergic Skin Disease Committee and one of the instrumental, together with Anne-Marie, who helped us to organize this session. Thank you very much. So please, doctor, go ahead.
Thank you so much, and I want to personally thank everyone from the IEC for proposing today's meeting and organizing and supporting. We really appreciate it. And yes, I am the outgoing chair, and Anne-Marie is the incoming chair of the Derm Committee here at the AAAAI. So I'm a clinician, and so my slides are very clinically based. I'm going to talk a little bit about some background from irritant contact dermatitis, because I'm guilty of doing this myself. Even though the majority of contact dermatitis is actually irritant contact dermatitis, we tend to focus and speak on allergic contact dermatitis a lot, specifically being allergists. So what's the clinical presentation of patients with irritant contact dermatitis? So irritants can be anything that the patient comes in contact with, include biologic sources, including saliva, urine, or stool. There can be chemicals found in personal care products, cleaning products. Our previous speaker, Dr. Atwood, talked about surfactants, but biocides as well. And it's any external agent that acts as a physical or chemical irritant to the skin and, in quotation marks, causing a non-allergic inflammatory response. Both irritant contact dermatitis and allergic contact dermatitis are going to have similar cytokines involved. It's just getting to the point where the cytokine involvement differs. For irritant contact dermatitis, there's no prior sensitization involved, and there's a propensity for the irritant contact dermatitis to include the face, the hands, and eyes. So this schematic is from Dr. Gutman's article about 7 years ago. This is a great article. It talks about atopic dermatitis, irritant contact, and allergic contact. And as I was saying, there's direct skin damage that's caused by the irritant that releases pro-inflammatory cytokines. And these are similar whether it's allergic contact or irritant contact. So you'll see things like IL-8, GM-CSF, IL-1, TNF-alpha. And these then will activate Langerhans cells, dendritic cells, and endothelial cells that will recruit lymphocytes, neutrophils, macrophages, and mast cells. And additionally, toll-like receptors and nod-like receptors can also activate these pathways. So irritant contact dermatitis is a complex picture. It can vary based on factors specific to the host, factors specific to the irritant, and some other environmental factors that I'm going to discuss. So the age of the patient, the sex of the patient, the body region, and the presence of atopy influence a person's susceptibility to irritant contact dermatitis. Additionally, there can be factors specific to the irritant. So the type of irritant, the amount of exposure, the duration of the exposure, the concentration, and the frequency also influence. You can see it in occupational and non-occupational settings. And in terms of occupational contact dermatitis, it's more frequently seen in patients who do wet work. And usually you'll present with a dry red scaly rash, like lichenified lesions. Usually the rash is more well demarcated and confined to the area of contact with the irritant, specifically if it's an acute presentation of irritant contact dermatitis. Now there are some, as I said, host factors. We see it more in men than women. In women more than men. We see it in children. Diaper dermatitis, even for the pediatricians in the room, is a very common manifestation of irritant contact dermatitis. We can see it in children who wear socks and occlusive shoes. We can also see it as a perioral dermatitis. And as I mentioned already, the face, the dorsum of the hands, and the finger webs are more prone locations. And atopic patients have a lower irritancy threshold that will allow irritants and contact allergens. And frequently we see ICD preceding ACD in patients with atopic dermatitis. So what about the environment? There's some factors particular to the environment. Cold temperatures can increase transepidermal water loss. Changes in humidity, particularly low humidity, can disrupt the skin barrier. Certain occupations can increase your risk of ICD, and this includes us, so medical and dental personnel, hairdressers, metal workers, food handlers, including chefs and cooks, homemakers, construction workers, and cement workers. The irritants themselves can vary. We can have things that are degreasing agents. We can have detergents. We can have dust, including stone and saw, food, and as I said, chefs and cooks, urine, saliva, surfactants, solvents, water, and wet work. And wet work, for those who are not familiar with occupational contact dermatitis, 2 is a big number here. So hands regularly in a wet environment for more than 2 hours per day. Frequent hand washing or the use of a hand disinfectant more than 20 times in a working day. or the use of protective gloves greater than 2 hours a day, or changing your gloves more than 20 times a day. There are chemical irritants, as I alluded to before, surfactants, disinfectants, antiseptics, and acids and alkalis can cause a pretty severe acute irritant contact dermatitis with ulcerations or burns typically seen in occupational settings. And then you have the physical irritants themselves, wools, textiles, and this we see very frequently in our patients with atopic dermatitis, having wool clothing, synthetic clothing, irritating the skin. And as I talked about before, the sweaty sock syndrome, so wearing woolen or heavy socks with occlusive footwear and sweating can cause an irritant contact dermatitis in the feet. So this was looking at patients who were patch tested, and so they had a lot of patients who were had either wet or dry work, and they patch tested them. And this was a retrospective review of over 1,600 patients over 10 years. And those that had skin barrier disruption caused by chronic wet work were an increased sensitization for these contact allergens. And again, Dr. Atwood alluded to lanolin being more of a sensitizer if your skin is already inflamed or affected. However, also those with, childhood dermatitis, which was interpreted as atopic dermatitis, were predisposed to contact allergy. So how do we diagnose irritant contact dermatitis? As allergists, we know history, detective work is our most common and most helpful asset. We want to know whether this is acute or chronic. We want to know if the patient does have a history of atopic dermatitis or if they have any other atopy. We want to inquire about occupational exposures, including wet work, the use of gloves, exposure to dusts, the temperature at work, humidity conditions, and whether there's a use of cleansers. We want to know if the patient wears personal protective equipment at work, and is there improvement, especially away from work. And we want to know about exposure to medications and query about personal care products and hobbies. Patients with irritant contact dermatitis typically will complain of also, besides itch, pain, burning, and stinging, more of a discomfort than necessarily itch that we see with allergic contact. And on the physical exam, some cues here. For eyelid dermatitis, allergic contact dermatitis will have both upper and lower eyelids bilaterally. However, irritant contact dermatitis on the eyelids is usually limited to the upper eyelids, centered on the folds, and can be about the cause of 15% of eyelid dermatitis. Here, allergic contact is the most common reason. For lip irritant contact dermatitis, you'll see some lip licking. We see a lot during the cold, dry months where patients are trying to increase moisture, but will actually wind up causing some irritant contact dermatitis. So you can see it around the upper and lower lip, and commonly, as I said, associated with lip licking or irritation from saliva. And then hand dermatitis, as Dr. Atwood also mentioned, presents as on the dorsum of the hand. Sometimes you can see some extension to the ventral surfaces and also the palms and the ball of the thumb. So the disadvantage of going and speaking later in the day is that some of this stuff has already been talked about, but we discussed already skin barrier dysfunction, correct? And so the compromised skin barrier in patients with atopic dermatitis, including decreased filaggrin or filaggrin mutations in certain patients of certain races, decrease in ceramides and antimicrobial peptides, increases in serum proteases, and here you'll see here you see very close, tight junctions in normal skin, and here you'll see disorganization due to filaggrin mutation, transepidermal water loss. And so this allows, again, irritants and allergens to get into the skin and to penetrate the skin layers. Looking at the presence of atopic dermatitis as a risk for irritant contact dermatitis, the risk increases 2 to 3 fold for occupational irritant contact dermatitis in patients with atopic dermatitis. And if there's the presence of both atopic dermatitis and filaggrin mutation, the risk to develop irritant contact dermatitis is 5-fold increased. Anne-Marie also talked about this a little bit, and our first speaker did as well, is that there's an altered microbiome. And here in the slide, you'll see sort of healthy skin that has a very diverse microbiome. The Staph aureus is the grapes in the pink color. The Staph epidermis is the grapes in sort of the blue color. And you'll see that with atopic dermatitis, there is a great increase in Staph, but you'll see during a flare, there's even a greater increase in Staph aureus and decrease in diversity of the type of organisms present. So this increase in Staph and other altered microbiome further compromises skin integrity and exacerbates inflammatory reactions. And then, as also was mentioned, immune dysregulation. So irritants encounter a Th2-primed milieu. You'll see increase in Th2 cytokines, increase in the Fc epsilon R1 receptor, increase in TSLP, increase in IgE, and this leads to sensitization. And as we mentioned before, lower irritancy thresholds. So even mild irritants, again, like Dr. Atwater already mentioned, lanolin can cause symptoms on already damaged skin. And so patients with atopic dermatitis have lower irritancy thresholds related to the skin barrier impairment. So when we're evaluating a patient for irritant contact dermatitis, we're thinking about things, the risk factors based on their work and their atopy. We're looking at things including wet work, occupation, and then we're looking at the agents themselves. And patients will present with erythema, they'll present with lichenification, they'll present with burning and pain and discomfort. And this is from death of the keratinocytes, inflammation, perivascular inflammation. So, takeaways, we said ICD is modulated both by intrinsic and extrinsic factors, influenced by things including age, sex, body region, particularly, as I said, the hands and the face, the presence of atopy and the irritant itself, the amount of exposure, the concentration, the occlusion effect, duration and repetition, and extremes of temperature, humidity also affect the influence of ICD. We've seen that skin barrier dysfunction, altered microbiome, and immune dysregulation all contribute to the atopic patient being susceptible to ICD. And thank you for your attention.
Thank you very much. And now we switch gears and we start looking into treatment options. And of course, we start with prevention, which is the most important one. Can we stop the atopic march? Are we there yet? I think that the next speaker deserves no introduction, just a brief mention that Professor Vanda Pipatanakul is the director of the Clinical Research Center at the Boston Children's Hospital and professor of pediatrics at Harvard Medical School. And she has been, instrumental in looking and preventing asthma and allergic diseases and led many cohorts. And we all learned a lot from her research. And we are very eager to know what is the recent views on this. Thank you.
Advance, we just use or this. Just
Either.
okay. Thank you so much for the organizers. This has been a bunch of really fantastic lectures. And this concept has been brought up a few times.
So it is the million-dollar question. Can we stop the atopic march? Are we there yet? These are my disclosures. And really, I think over the next 20 minutes or so, I'm going to review a little bit, kind of summarizing a bit what has already been mentioned as the armamentarium for atopic dermatitis, and can we do anything to halt the atopic march? I'd like to start with a case. A lot of us are clinicians, right? And this is a patient that we most likely have often seen in our clinic. We have a 2-year-old male who comes in with a history of eczema and milk allergy. He was just evaluated in the emergency room with a really bad case of wheezing, and he has a family history. Dad and brother have moderate persistent asthma and allergies, and mom has bad hay fever. And this comes in my clinic all the time. Mom is very concerned, and she says, Dr. P, or Dr. Wanda, depending, will my child develop allergies or asthma just like all the others? Can I do anything? Is there anything at all that I can do to stop this process? How many of you heard a question like this in your clinic? So I'd like to remind you of the allergic atopic march. I think this has been shown a few times from many of the speakers. And this is like our bread and butter, right? Those of us allergists, we see those kids that come in with food allergy and eczema into the clinic, and they go on to develop allergic rhinitis and allergic asthma. And people are even saying we've got a new thing on the block now, eosinophilic esophagitis. And these are really driven by IgE. Think of us as allergists. What would we do without IgE? Kind of out of a job, right? So really, the allergic atopic march is really important to think about, and it just starts young. What do we think about atopic dermatitis in treatment and prevention? So this was a review that was just published last year by the Journal of the American Academy of Dermatology that reminds us the first line is always moisturize, right? We do the hydrolatum, petroleum jelly, moisturize, moisturize, moisturize. And then you've got your topical therapies. We have our mainstay of topical steroids. We have now some new kids on the block, the phosphodiesterase 4 inhibitors, the JAK inhibitors. How many of you are starting to use those? You've got the wet dressings. We go and hang out with our dermatologists. I'm glad to see there's another dermatologist giving these talks. We do sometimes phototherapy, systemic therapy. And then we have the age of systemic therapies like biologics. Wow, how exciting was that when Dupy became FDA approved for atopic dermatitis? And now we've got a few more kids on the block. Some of the anti-IL-13s have recently been FDA approved. And also, as you know, Dupy is approved down to 6 months for atopic derm. Really, really exciting stuff. We've got the JAK inhibitors. I know Sharon and some of the others started talking about those. I will not try to pronounce those as well. They're harder to say than my last name. But JAK inhibitors are also some new ones to think about. Do we think about it in prevention? I mean, there's always a little concern about questionable safety. It's really more approved for the older kids. And then you've got other stuff like methotrexate, cyclosporin, and then systemic corticosteroids as well, which most of us try to stay away from, right? How often do you guys want to do that? I always find I do it, they get better, and then they really flare even more. So look at what we have. That's a lot, right? The field of atopic dermatitis and these therapies have really exploded in recent years, making it very exciting to think about that we have more treatment, and then could we possibly prevent or modify disease progression? This was a review that was done by one of my fellows a couple of years ago and just kind of highlights again some of the targets, some of the areas that we are thinking of. In addition to all the new therapies that have come out, are there some other pipeline molecules? Someone had showed some data about TSLP that might be of interest. There's this OX40 ligand. We know that the therapies, a lot of them are already FDA approved for other conditions, such as asthma. We got a bunch of anti-IL-5s. Of course, we have Dupy. We have the anti-IL-13s. We have omalizumab, of course, the first biologic that came out 20 years ago. And now even in the horizon, maybe some hyper-IgE, anti-IgEs, and anti-IL-17s as well. And all of this armamentarium is a really interesting time where we as allergists and immunologists, we're like on the forefront, right? We are using these therapies. And when it comes to atopic dermatitis, you can see how this really all comes into play. The immune system works. We've got that kid or adult who's scratching their atopic dermatitis. You have that kind of itch scratch cycle. And then you have allergen exposures. You have microbes. Someone mentioned about Staph aureus. And then it goes into the immune system and you really rev up that process of atopic dermatitis. And that's that first pathway that goes on to the food allergy, to the rhinitis, and the asthma. And if we could block these processes, and that's what makes it so exciting as allergists and immunologists, that we understand these pathways. We see these diseases across the pathways, and can we make a difference? So what about targeting kids with eczema and the atopic march? This concept has been around for a while. I'd like to show a couple of studies that might show some interest. And I saw that Sharon's doing a couple of studies looking at moisturizers and food allergy, which is really exciting. This is a study, it's pretty old now, it's almost 10 years ago, where they took about 60 infants that were high risk of developing atopic dermatitis, and they just moisturized them. So simple, right? Moisturizer versus a control element. And they followed them over the first 32 weeks of life. And what you can see is that it did look quite promising in preventing the development of atopic dermatitis. So that started to get people, can you do something that simple and prevent the atopic march? Well, there have been lots of studies that have been looking at moisturizers in prevention. I will be very interested to see what it does for food allergy, Sharon. So I saw a couple of those studies. But moisturizers in prevention for atopic dermatitis. So what they did, there's a Cochrane systemic meta-analysis or systemic reviews. We see those, right? Where they take all the studies. There's lots of limitations with them, right? Because you can't really combine all the data. But it was done in 2022. It looked at 11 randomized trials with over 5,000 participants. And they looked at moisturizing in infancy. And what they found, and then I saw in some of the previous slides as well, that it really did not change the risk of developing atopic dermatitis by age 1 to 3 years of age. They looked at other factors as well, the type, the duration of the emollient use, whether they had that filaggrin mutation or a chromosome and risk variant, whether they had family history of atopy, and that did not seem to impact the risk. The skincare interventions, actually, when they looked at the data using the emollients, thought that they may show some signals of actually increasing the risk of developing an IgE-mediated food allergy by 1 to 3 years when compared with standard of care. Now, I'm sure there are limitations in these studies, and I'm sure there are many factors and biases related to that. But so this Cochrane data review says the use of emollients may actually increase the risk of skin infections for whatever reason, and it may not be that a beneficial strategy in preventing the atopic march. Now, there are two approaches that have been well published, and I know that there are other studies going on, super exciting to have all of these prospective trials, where they took kids with eczema and tried some therapies. One of them is called the Study of Atopic March, the SAM study, where they used a calcineurin blocker and gave them to young kids and saw if they could prevent the atopic march. Well, there's a main figure from the paper. What do you think? Did it work? No, really disappointing. Compared to control, you really see no difference at the beginning of the double-blind placebo-controlled application of the Elidel, basically, the calcineurin blocker, and then at the end of the study. Really no difference. What about using something like an antihistamine, something simple like cetirizine in young kids? Super easy to use and follow them for years. They're going on to develop eczema. Maybe if we can stop them from scratching, could that prevent the atopic march? And, well, they did the study, and what do you think? Disappointing, right? So kind of disappointing that really at least these two approaches in kids with eczema did not prevent the atopic march. What about probiotics? Do you guys recommend for your patients probiotics? Could we alter the gut microbiome and prevent eczema or asthma? So this is a study done by Michael Campana back when he was at UCSF. He's now in New York. And he looked at a double-blind placebo-controlled trial using lactobacillus and inulin versus inulin alone in 100 high-risk infants. This was an intervention at the first 6 months of life. He's a pediatrician. And what you can see is he then followed them to the cumulative incidence of asthma. What he found is at age 2, kind of disappointing, no difference in eczema. And at age 5, I talked to him about this study. It looked so many different ways and really no difference in asthma outcome. So in general, most of the data has been not supportive for probiotic uses in preventing the atopic march. And then not recently now, it's kind of old now, but in 2016, they did a meta-analysis looking at 2,403 articles, 29 studies, and their conclusion was supplementation of probiotics during pregnancy in breastfeeding moms or during infancy may reduce the risk of atopic eczema. Okay, so that's good. But the certainty is very low due to the bias, inconsistency, and body of most of the evidence was indirect. It also showed that it does not reduce the risk of other allergic diseases or asthma in children. I think there's no huge downside to using probiotics, but the studies have not been completely supportive that they will prevent the allergic atopic march. What about microbe exposure? We all know, right? We've learned that kids growing up in the farm, they have less asthma, right? But how easy is it to move all our patients to the farm? I live in Boston, it's like about impossible. So there's been a lot of interesting data, lots of press understanding about the microbiome, that hygiene hypothesis. Is it possible that we're now too clean? We're not exposing our kids to microbes and dirt? And are we really causing asthma and allergies to increase in prevalence? Is that why there's more food allergy? So I'd like to say that we do also have another big NIH-funded study that's really looking, could we alter the microbiome? You know, these kids that they live in farms, they have less asthma. Can we introduce something that could alter that microbiome in prevention? So there's a study that's ongoing called the ORBEC study. And what we're using is there's an oral killed bacteria lysate, which is a cocktail made of a lot of extracts of bacteria, basically killed bacterial bugs, includes H. fluenza, Staph aureus, Klebsiella, lots of other things. It's in a substrate. And it is actually in Europe. Anyone here from Europe? It's available over the counter in Europe. It's actually, many times what I've told from my colleagues is, if your child has a cold and they go see the pediatrician and they don't want to give antibiotics, they're like, just take this. It's over the counter. It's a killed bacterial lysate used to treat the common cold. It is not live bacteria, so it's not the same as the probiotics. So this is available in Europe. It's not available in the United States. And it's been marketed in the European Union for the prevention of respiratory illness for over 25 years. Those are from Europe. Do you think it works? No. Okay, all right. So we got a study funded through NIH. Fernando Martinez is the lead. We are one of the sites in Boston. There's about 10 sites around the country, all over, including the West Coast and the East Coast. And it's called the ORBEX trial. We have already recruited 800 babies, and they were recruited at age 5 to 17 months. They had either eczema or a family history of asthma in a primary prevention study to see, can we alter this gut microbiome and can we make a difference? So we are done with recruiting, and I think it's late 2026, we will have the results of this study. So basically, we treated them early on in life with 2 years of this cocktail of killed bacterial lysate, altering their gut microbiome, and then we're following them until they're old enough to see if we can determine asthma. So stay tuned, really exciting, and hopefully we'll figure out. It seems to maybe not work so great for the common cold in Europe, but maybe it will have something to do with asthma or the atopic march. So what about another concept? We're allergists, right? Remember I showed you on that first slide how important IgE is to our specialty. So what is the triple threat when we think of the atopic march and asthma? What are 3 factors that we think are so important? We're allergists, right? We know that atopy is a pivotal risk factor in the development of asthma. It's been found actually that if you're atopic and then you have this viral wheeze, your risk of asthma just skyrockets, 80-90% in many cohorts. Of course, viral wheezing is important as well, but not all kids that wheeze with viruses end up developing asthma. It's really those risk factors, right? You're genetically predisposed, you are often atopic, and then you have viral wheeze, and you go on to develop asthma. What about exposure? Okay, exposure also can predispose you. You're allergic, you have IgE-mediated disease, and then you start wheezing around a cat, you get some peanut, and you're allergic to peanut, and any exposure that you might have may also be, what I view as these 3 factors are just the triple threat when it comes to the development of asthma in children. So can we hypothesize that an early intervention in young children, when they're kind of going down that path, remember that kid I talked about on the first slide? You know they're going down that path. Could we intervene on something that is feasible, that acts on the triple threat that could possibly prevent the atopic march? And I'd like to hypothesize that hopefully we can. And that's what some of the premise is behind the PARC study. It's preventing asthma in high-risk kids. It's a double-blind, placebo-controlled trial using something that has been around for a very long time, that we could convince the FDA that it's safe to use in young kids as a prevention strategy. So the kids are enrolled, and they are high risk, really high risk of developing asthma and the atopic march. And we're treating them for 2 years with either anti-IgE, omalizumab, versus placebo, and then we follow them 2 years off therapy, and there are 2 primary outcomes. The first one, did we prevent asthma? Home run, right? Grand slam. Did we prevent asthma? The second primary outcome in the study is in those children who develop asthma, could we modify the progression of the disease? Could they have less asthma severity? So in either way, we really could make a difference in preventing outcomes. We're of course looking at all the other allergic outcomes. We're looking at eczema throughout the trial, wheezing, rhinitis, food allergy, and we have a host of mechanistics as well that are planned and funded in this study. So we hope 2027 is when these studies, each child is 2 years of treatment and 2 years of follow-up. As you can imagine, recruiting a study for a double-blind placebo-controlled trial throughout the pandemic has taken time. So everyone is asking me, when are the results coming? Patience, but 2027, we will have a read for the outcomes of this study. And so we hope, and this is some websites on the study when it first came out, and we are now funded, of course, through NIAID, hoping lots of support continue with the NIH to allow us to complete the trial, and then also have lots of really interesting mechanistics planned for this study. So people always ask me, what about Dupy? All right, Dupy now is FDA approved down to 6 months of age. One of the challenges in doing a study like this, in a prevention study, is once it's already approved for the young age, it's very, very difficult to do double-blind placebo-controlled studies in very little children. In addition, also, it's been hard probably to have, what I've heard is that the company does not really want to support long prevention studies such as this. So we've got the PARC study, but people do ask about Dupy. But this is some of the data that we have looking at lots of clinical studies. And when you compare Dupixent versus placebo in many of the allergic outcomes, looking at the allergic march, you can see the risk ratios. Not overwhelming, but again, I think there's lots of limitations when it comes to these meta-analyses. But looking at it, you can see some potential benefit, but I think it will be very hard to do this type of study, similar to a PARC study in dupilumab. People have talked about it, but people do always ask me about that. But still, we'll have a lot of data. Kids who are being treated routinely for eczema with it will be able to understand and follow outcomes. And I think also PARC was developed for years prior to this, but doing prevention studies in things not only that they're FDA approved, but long-term safety. We're getting more and more, but Dupixent is still relatively new on the block, especially the number of young infants treated with Dupixent is actually pretty small. But stay tuned. I think it's an exciting time, but I had to show a little bit about Dupixent as well. So in closing, prevention, the holy grail, right? Really, we are at that time now. It's an exciting time. The landscape of treatments in atopic dermatitis and asthma and in our field has really markedly increased. It's been incredible. There have been lots of strategies that have been attempted, but really nothing has proven to be effective in modifying the atopic march. There are two really exciting NIH studies, really long-term studies targeting microbiome in the Orbex study and atopy in prevention. These are a lot of effort, as you can imagine, take a lot of patience, but we are going to have some readouts in the next couple of years. Newer immune therapies, we really need probably more safety real-world data before the FDA is going to let us to use a lot of these newer therapies to consider it as a treatment or as a prevention strategy. But stay tuned, and if they're already FDA approved, it does get harder. I thought, wow, Dupixent got approved so quickly down to 6 months. It's hard to do a double-blind placebo-controlled trial for this type of question, and placebo-controlled trials that you can imagine in biologics, not the easiest thing to do. But I think our advancements in understanding what may work in prevention and modifying asthma and allergies, we're there. In a year or two, couple of years, we are going to have some readouts from some really big NIH-funded studies, and it will be exciting to see, because no matter what, we will learn so much about what happens early on when you've got that kid who's going down that allergic path. Can we do something to prevent or modify disease progression? Sign me up. I would have done that for my 20-year-old kids, and I hope for next generations as well. I'd like to really thank NIH and all the regulatory agencies that have been supportive of the study, all the sites that have been involved in this study. And this was really inspired. My late mother was an allergist, suffered from tons of allergies and allergic diseases. I have also passed that on to my kids. And I hope through learning from the PARC study that maybe my grandkids will not have to suffer anymore from these diseases. So thank you very much. And at the end, we'll take questions.
Thank you. So there is hope on the horizon. Let's not give up on good research. And then we go ahead to what is currently in the pipeline with the novel therapies for atopic dermatitis beyond what we are currently using, IL-13 and the JAK inhibitors. And it's my pleasure to introduce the next speaker, Dr. Raj Chovatia, Clinical Associate Professor of Medicine at Rosalind Franklin University, Chicago Medical School, and founder and director of the Center of Medical Dermatology and Immunology Research. Please.
All right, thank you very much. And that was perfectly timed, because we now get to talk a little bit about exactly what is coming and what is happening, and this amazing idea of disease remission, modification, changing something about the disease may not necessarily be as far away as we think. So these are my disclosures for a lot of work I've done on many of the things involved in this talk that are actually very, very exciting. And since you've spent this whole day hearing from people much smarter than myself about the intricacies of the science behind the disease itself, I'm going to maybe skip the review of exactly what is atopic dermatitis, but I tried to divide up everything we're talking about today in my session on the themes. In particular, what I want to really talk about is this idea of what are the different places in atopic dermatitis pathophysiology we can look to really try to treat the disease perhaps differently than targeting IL-13, JAK inhibitors, and some of the downstream cytokines or signaling pathways that we've learned about. So if I was to group these, I would say we're going to be looking upstream. We're going to talk about maybe focusing on the keratinocytes a little bit. We're going to try to get inside the cell in a different way. We're going to talk about how we can maybe rebalance our immune response as another strategy. We'll touch on cell trafficking for just a moment as a cautionary tale. And then my last section about being everywhere and everything all at once. Okay, so let's talk about moving upstream. This is where we're at with atopic dermatitis at the moment, right? We've tried to really understand what individual cytokines are doing, get down to the level of JAK-STAT signaling in terms of JAK pathways. And, each of these options has its own limitations of not only efficacy, but safety and ability to really broadly treat this disease itself. And it's one reason why maybe we'll never really be able to, with our at least current therapies, understand this concept of remission or prevention, just because maybe we're operating too downstream in an immunologic pathway. So naturally, what if we looked up and thought a little more broadly about what's happening in this disease state? And I know sometimes this idea, especially with the advent of JAK inhibitors, and it's a different time for different discussion, people think broad equals bad, but I'm here to make you think that maybe broad isn't as bad as you might think. So, we haven't really figured out how to treat atopic dermatitis, obviously, as early as possible for the clinical signs of disease itself. But what if we focused on a time period where we were thinking about some of the earliest signals that are happening in keratinocytes, things that they're producing, the earliest interactions between T cells and antigen-presenting cells. And while some of those early stage cytokines have been a bit of mixed picture, some of these trials did not hit the way we wanted to, what if you perhaps targeted T cell immunity in general, one of the earliest things that's required for a heavily T cell dominated disease? And that's really where the story of OX40 and OX40 ligand comes in. And that's going to probably dominate about half of my time here with you, just because it's what you will be hearing about nonstop. You're going to be hearing about this meeting. You're going to be hearing about it at a lot of meetings over the next couple of years, just because this is sort of the next big frontier in a lot of different inflammatory disease states. So what I really want to go stepwise is maybe help you understand exactly what OX40 and OX40 ligand are for those of you guys that maybe haven't been too involved in what's happening in the science, to give you a better idea of why this makes sense for a disease like atopic dermatitis. So when we think about some of those steps that are involved in disease pathogenesis, and we know that AD is inside out and outside in, but just follow me here a little linearly. We know that there's events happening with the barrier being perturbed, things activating the immune system, causing a cascade downstream. T cells end up getting activated, polarized, differentiated, and turned into long-lasting memory cells. And you have a lot of downstream effects on both the keratinocytes, the epidermal cells, of course, neurons, and immune cells too. And if we focus in on sort of looking a little bit to the left of this diagram, right around it says 1 and 2, you'll see that this is where the antigen-presenting cell and T cell are having their first date and meeting, and this is where the story of OX40 and OX40 ligand begins. So it actually really occupies a pretty important central role in the disease state itself. So when we think about what's going on with this interaction, take ourselves back to thinking about sort of immunology 101, we know that with an antigen-presenting cell and a T cell, we need signal 1, signal 2, right? Typically, we need something to activate the innate immune system. That's going to kick a few things into the gear. Then we know there's going to be MHC processing, there's going to be presentation of an antigen and binding of a T cell receptor. But that isn't the entire story, because we know that alone is not sufficient to do all of those wonderful things I talked about that T cells are capable of doing. And you actually require a number of co-stimulatory signals. So some of the ones that we think about all the time are like B7.1 and 2, it's CD28, that's one great example. There's others, there's CD40 and CD40 ligand. You can think about OX40, which is a receptor, and OX40 ligand, which is a ligand, as a secondary co-stimulatory interaction that is not there at baseline, but becomes activated upon the presentation of MHC and T cell receptor together. And it's really important for those two to bind to each other in a lot of different states of T cell activation for many of those features that I mentioned in terms of not only just activation of the T cells themselves, but quite importantly, their eventual polarization, proliferation, and formation of memory cells itself. And so you can see here that just again, at baseline, typically you're not going to be having OX40, which is expressed on T cells, OX40 ligand, which is expressed on antigen-presenting cells. I'll say that again because it's kind of hard to remember, but if there's anything to take away that part. OX40 itself is on the T cells, and OX40 ligands on antigen-presenting cells. OX40 ligand comes up a little quicker, takes a little longer for OX40 to come up, and that interaction is sort of key to a lot of the next steps we think about in the T cell-mediated immune process. So there are multiple signs and scenarios, both in basic science and even translational science, that are shown that OX40 and OX40 ligand have a sort of important role in both peripheral and central immunity. We know that there can be increase in OX40 positive T cells as much as tenfold in lesional skin for patients with local inflammation. We know that in skin biopsies of atopic dermatitis, there's co-localization of these two things as well. So there's definitely a peripheral immune interaction. But there's also a central immune interaction as well. We know that circulating T cells have elevated expression of OX40 itself. And when we kind of follow along what's happening, I showed you a slightly different version of this diagram, but perhaps it kind of makes sense now that you're going to be having something happening with keratinocytes, some of your earliest alarmins and cytokines being produced. You're going to get activation of your antigen-presenting cells to present something to T cells. And OX40 ligand and OX40 are really that link there that then results in your production of Th2 cells, Th1, Th17, their high degree of activation, and even the formation of long-lived memory cells too. And so for a disease like atopic dermatitis that's inherently quite heterogeneous, and we know that simply just targeting one branch of immunity, like with IL-13 or 4 and 13 blockade, can work, but it's not going to be 100%, and it's not going to cover everybody. We also know that with atopic dermatitis, it can be a hard disease to diagnose dermatologically because it can look so different from person to person. And so there's always been this idea of an underlying immunologic heterogeneity, and there's sort of some, but it's not the most amazing, but some evidence that points us in that direction. And so really this type of target is one that can really test not only that hypothesis of a heterogeneous immune interaction, but one that may actually have longer lasting effects because of its ability to not just hit a downstream cytokine, but actually change what's happening in terms of the memory compartment and formation of memory cells. And you can see here that there's multiple molecules in development. The two in which are furthest along we have the most data for. On the left, you see amlitelimab. This is a non-depleting OX40 ligand antibody. And on the right-hand side, this is rocatinlimab. This is a depleting OX40 antibody. So two different strategies. One that's really designed to work on the antigen-presenting cell side of things. One that is designed to deplete the pathogenic T cells. And so it is kind of an interesting immunology question about which one of these is going to prove to be perhaps the most effective strategy. And there are others in development here as well. And so that's where our discussion begins. And so we have phase 2 and some preliminary phase 3 data on rocatinlimab. And next week actually is going to be a late breaker at the American Academy of Dermatology meeting where we'll hear all about what's happened with some of the recent phase 3 results with roca. And then we have phase 2 data with amlitelimab and phase 3 data not too far behind as well in the future. So this was the general setup of the published phase 2b study for rocatinlimab. A couple of differences perhaps than what you're used to maybe with some of our other studies in atopic dermatitis. This one was taking a look at a primary endpoint of about 18 weeks. This is a slower mechanism of action. You'll see this in the data that I show, and it's possible that just the endpoints with which we're so used to using may not be the right ones to look at this particular lens. There was also a treatment period B, which you can see here, going to 36 weeks, and then a follow-up period that allowed for people to actually be off drug, and that's perhaps the most important part of this study. So when taking a look at what happened in the actual results, you can see bottom line, you're looking at delta EASI on the left-hand side compared to the placebo at the top. This shows what phase 2 should show. There's definitely a difference. It's in the ballpark that we'd expect typically for something in phase 2. It says that you have a mechanism of action. And the coolest part was if you look all the way on that graph on your left-hand side and just follow it to that right section where it looks kind of pink, that entire area is actually a follow-up period off drug. So going from week 36 to 56, those are all people that actually had all treatment with roca removed. And if you take a look at that group on the right-hand side in that Kaplan-Meier curve, this is looking at time to loss of EASI 75. fascinating, but really it's only in the lowest dosing arm that you're really starting to see some substantial drop off, perhaps after 20 weeks. But by and large, people are doing pretty well in terms of the endpoints that we think about. And now, this doesn't have to be a debate on whether this is the right endpoint to use or the right time to use, but I think it is very, very intriguing since this is not something we're typically used to seeing, especially even at this stage with a systemic atopic dermatitis therapeutic. I'll mention from a safety standpoint, largely things look promising. The one safety signal to watch here, chills, pyrexia, and nausea, we're all seeing at measurable rates and will be the major thing to follow. Unclear if this is related to perhaps the actual mechanism itself or the drug. And this will be an important differentiator with when I talk about amlitelimab in just a moment. So this is some of the preliminary data that was released with rocatinlimab. This data actually was just released in a press release, investor presentation, actually right around the time of EADV, the European Derm Academy meeting just last year. And so this is what we're going to get some more context on. So this is taking a look again at monotherapy over the course of 24 weeks, primary endpoint. You can see here some of the EASI-75, VIGA, and another modified IGA result overall. The main takeaway is that perhaps these deltas slim down a little bit from what we saw in phase 2. And I think that these are probably a little skinnier than we're used to seeing at this particular time point. Now, we don't actually have that much context as we know that there was bio-experienced patients that were included in here. So this is pending full presentation. We don't actually have full information on the dose itself as well too. But bottom line, all the endpoints met statistical significance. And I would tell you that, in this particular mechanism of action and even OX40 ligand, I'd say that your set point of probably thinking about what the ideal efficacy level should be perhaps might be different since we're really trying to answer a slightly different biological question. So this was the design of STREAM-AD, the amlitelimab phase 2b study, which was recently published in JACI at the end of November. And you'll see here that there was a lot of different dosing regimens looked at without loading dose and with loading dose both. And then there was a rollover period into part 2 where individuals were allowed to go into a withdrawal period where they weren't on drug or they were continued on maintenance dosing overall to essentially look at the same idea. That is this a drug that is truly causing something that's modified about the immune system in order to allow people to take these types of breaks. And so again, this drug, remember, different. This is OX40 ligand as opposed to OX40 itself. You'll see here in terms of looking at our delta EASIs, this is phase 2 data overall by week 16, week 24, you're kind of starting to see that difference. Again, these are in the ballpark of most of our systemic therapies, maybe a little slimmer overall, but still kind of right around where we should expect to be seeing a signal for something in phase 2. But the interesting data, again, comes from this looking at withdrawal period overall. So this is just pooling all the way across the doses on the right-hand side at week 52. And this is taking a look at the responders and then what happened to them over that period. And what you really want to pay attention to is the gray bar, which is the placebo withdrawal, and then the red bar, which is the continued amlitelimab. There's two analyses on this chart. One is an NRI and one is all comers. But the bottom line is that these look also pretty similar as well, suggesting that, again, the pattern seems to be true for this interaction between OX40 and OX40 ligand. It appears that we're really able to actually push the periods in which people seem to be able to be off drug. The off-drug efficacy actually was confirmed by biomarkers as well. These continue to stay low. So things like TARC, IL-13, 17, and 22 were all looked at. And this was also very encouraging as far as safety signals overall. Just to prove a point, this is actually taking a look at the active continued arms versus withdrawal arms, and you'll see that overall drug levels also dropped below detection as well too, suggesting that this wasn't just due to very long half-life continued circulating drug in the systemic circulation. I'll touch on one more that was recently presented. This is an OX40 targeting drug, though this is a little different. It does not have the FC sort of domain that causes antibody-dependent cellular cytotoxicity. This one has a mutation there. So unlike rocatinlimab, this does target OX40, but this one is non-depleting. So yet another variable added in terms of how this might look. This is just proof of concept phase 2A, but you can see here that bottom line, after just a couple of doses at week 0, 2, and 4, patients actually had measurable efficacy at week 16 and even 24. And so perhaps it's a little optimistic, but the conclusions of this particular poster presentation said they really think that they're going to be able to push extended dosing intervals with this particular mechanism of action. Okay, so for the second half here, I'm going to now go on a little world tour around the immunologic perspective of atopic dermatitis and talk about all the other things that are happening pretty early on. Most of these are phase 1, phase 2, and some of these have not even had phase 1 data released. So let's talk about keratinocytes. IL-22 is one target that perhaps had some disappointing results when we had a trial taking a look at an antibody binding to IL-22 itself. IL-22 makes sense in this disease. We know it's an important part of barrier disruption, epidermal hyperplasia. We know Th22 cells are involved. And the IL-22 receptor, IL-10 receptor sort of combination is actually expressed right at the border tissue between the host and the environment. So IL-22 is quite important when it comes to thinking about environmental signals, and a great example of why the aryl hydrocarbon receptor, which is one of our detectors of homeostasis, is actually an important upstream factor that drives some of this IL-22 production. So temtocobart's an inhibitor of IL-22 receptor A1. This is different than a previous IL-22 targeting molecule in that this is looking at a shared receptor subunit. So this actually is functionally inhibiting IL-22 as well as IL-20 signaling. And this was proven in some basic science in vitro assays overall. And so that's suspected why perhaps maybe there was a signal seen. And sure enough, you can see here in this phase 2A proof of concept study, that took a look at patients, maybe that fell in a little bit more of a moderate range, because it was moderate to severe. There was very clearly a delta here in terms of baseline EASI score at week 16. So an interesting one to watch and follow for the future. IL-18 is an interesting story. This one was recently presented with phase 1 data. And the reason why I say interesting is we've had a lot of lack of success in the IL-1 family with atopic dermatitis. IL-25, 33, IL-1 alpha, these are also all just alarmins too, but early phase cytokines have not fared super well in some of our trials. And particularly in terms of the IL-1 family, it's been a bit of a mixed picture too. Now, this isn't traditionally thought to be a driver of atopic dermatitis. For those of you guys that are familiar with IL-1 biology, you know that IL-18 is produced downstream of the inflammasome itself, so it's produced as a proform based on usually some innate immune signal, and then inflammasome activation, typically to something that's going to alter the barrier, is going to cause cleavage of the active IL-18 itself, and it's going to signal then through generally an IL-1 family receptor molecule. But IL-18 transgenic mouse models actually show spontaneous disease that's very, very similar to atopic dermatitis, and it's a reason why there was always hope there potentially that this might be just a pleiotropic cytokine that exists upstream in this particular disease state. So GSK 1070806, it doesn't have a sexy name yet, but this was a phase 1 molecule that was studied. And you can see here over the course of 12 weeks after just a single IV dose is what they were taking a look at with delta EASI overall. And so bottom line, there was a signal here after just that one dose. And so you can see here, both in terms of improvement in EASI score, itch improvement, dermatology life quality of index, you are looking at 20 versus 10 patients. So just take that with a little bit of a grain of salt. It's actually really interesting that the primary and secondary endpoints have been met. The next phase is moving ahead, and it's really going to be fascinating to speculate if this can make it to phase 3 as such an upstream cytokine overall. All right. Let's go back inside the cell, but not a JAK, since that was literally half my title, not JAK inhibitors, all right? So IRAK4 is a target that you guys might be familiar with, and it exists as part of the MyD88 Essentially, MyD88 is downstream of toll-like receptors and IL-1 as far as signaling goes. And we know that IRAK4 is an important kinase that not only has kinase function that allows for signaling to get activated for MAP kinases and NF-kappa B, it has scaffolding function as well. And previously we had a kinase inhibitor of IRAK4 that did not go the way that we wanted it to, but there is a molecule KT-474 that actually is a degrader. And so when I say degrader, there's two moieties on this little molecule. One is able to actually bind to the IRAK4 itself. One is able to tap into the ubiquitin-mediated proteasome system, basically at the step of E2 ubiquitination. And essentially, you're dragging that piece of scaffold out to get degraded. So in addition to stopping it from doing its kinase function, you also don't allow that entire signaling complex to be built. And so there is a lot of rationale for scientifically why this makes sense across a lot of different inflammatory diseases. And there was a phase 1 study that was published in Nature Medicine about a year ago, both in HS and AD, it was combined, that showed that there was degradation detected in whole blood, reduction in tissue markers of inflammation, and no drug-related signs of infection, which was really, really promising, because that is the one thing you could think about that might be worrisome here. And from the few patients they had, there was efficacy signal of some sort. So stay tuned for that particular MOA. STAT6 is another one that takes a slightly different approach, where IRAK4 perhaps is a broader part of an immune cell activation. STAT6 is fairly specific for downstream of IL-4 and IL-13 signaling. And so there's been a couple of molecules in development, one specifically inhibitor of the SH2 domain, one that's a degrader. They're all trying to leverage some of the similar technology I mentioned to saying if you take STAT6 out of the game, maybe this is a better way to do what JAK inhibitors are so successful at, but not with some of the other off-target effects that we may expect to see. And there's a lot of reasons to believe in STAT6. I mean, I don't need to tell this audience that individuals who have STAT6 gain of functions and mutations have severe allergic and atopic disease. And so inherently, I might have you there already. And so I'm pretty curious to see how this one fares as well. There's been a lot of work in rebalancing the immune response, right? We've seen this across a lot of therapeutic areas, but dermatology has not been spared from it. And in atopic dermatitis, there's been a couple of ways people have tried to take bites of this apple. So one is a very interesting drug called Respeg. It has a much longer name, Respeldaslucin, but essentially it's a polymer conjugated recombinant human IL-2 protein. And the idea here is that with its pegylation, it has much more specificity of Tregs without activating effector T cells. And the idea here is if you can actually boost your Treg signal, perhaps in some of these heterogeneous inflammatory diseases, not just atopic dermatitis, you may be able to make a dent in a different way broadly, kind of like we talked about with OX40 and OX40 ligand, but again, a little different since we're talking about the balance of immune activity. So this was a phase 1 study that we just published at the end of 2024 in Nature Communications, taking a look at a couple different doses of Respeg and placebo. And you can see here, bottom line, very few patients overall, but there was a period where people who were responders above EZ50 were allowed to just follow up after they were treated with drug and they weren't on therapy at that point in time. So this is kind of what the data showed. Bottom line, there was a change from baseline of the EASI score at week 12, so that's good for a study this early. But most importantly, when you actually take a look at this follow-up phase in the graph on the right-hand side, even though the numbers are quite small because we were really only looking at responders over this long period of time, what you'll see is that there's, again, long-lived responses for individuals off drug as well. And it seems like perhaps this coincides maybe with a spike in CD25 bright Tregs. If you take a look at facts, that's not necessarily long-lived throughout the study, but it's really just a transient signal. So the question here is that, is it just enough to really sort of set that off, and you don't need to even sustain that entire process to really change what's happening overall? And so this is continuing forward in development, not only in atopic dermatitis, but other disease states as well, and even in dermatology for alopecia areata. One study that perhaps is a cautionary tale, just because I don't want to give you the sense that everything works, ANB032 is a humanized monoclonal antibody targeting BTLA, right? And there was a lot of excitement here because there was a phase 1 that said that there was tolerability, safety, maybe there was some efficacy here too. And we know that BTLA is important for regulating T cell activation and proliferation. And so there was a study that was designed to take a look at a few different doses. Unfortunately, in December, it turned out that the absolute response rates while they were getting there just were not met. And so this one was pulled from further development, suggesting that maybe this is not the right approach to take as far as MOA. But one of the main issues here was that there was actually a really high placebo rate, particularly in the US. And this is a trend that we're starting to see in other atopic dermatitis trials too. So something to keep in the back of your mind when you start to hear some new data coming out about some of these other drugs. I'll just touch on J&J, another long number, 674-84703. So this is a humanized antibody that inhibits PD-1. I think we all get the idea of sort of how PD-1, PD-L1 work, especially from the oncology world as well. And so RESTORE-AD is a phase 2A proof of concept study design. I don't really have any data to touch on, but it's worth noting again that we're really exploring a lot of different nodes for balancing of immune activity. Cell trafficking, okay? So this one doesn't necessarily have a happy ending as well. There was a drug called zelnecirnon, or RPT193, that was actually looking at blocking CCR4. Now that one makes sense, right? Because we know that TARC and CCL17 and CCL22, two signals that we sometimes use as markers for atopic dermatitis, signal through this pathway. And it makes sense that if we could maybe change trafficking, this is a safer way than broadly inhibiting, let's say, JAK inhibitors in an oral package that would allow us to have the response that we're looking for. And so the trial was quite promising in terms of the phase 1B, that there was clear efficacy. There was actually off-drug efficacy, which was interesting too, and no one had a clear explanation for. And so then there was a phase 2 that set forth. Unfortunately, the phase 2 was terminated early with only about half of the patients having completed 16 weeks. This happened about last year. And as it turns out, there was liver failure in one patient that probably had a really long story for why they should have had liver failure. And it's hard for me to sit here and speculate this was drug related, but that was enough. And so this drug has sort of stopped further development in atopic dermatitis. So this one may not be the answer, but I still think CCR4 as a pathway has some interest, at least in the back of my mind. This one is just purely fun and not really data related. There are so many multi-specific antibodies currently in development. You literally can't throw a stone far enough and hit multiple companies that are looking at buy and try specific technologies, trying to target IL-4, 13, 31, TSLP, OX40, and others, either 2 at a time or 3 at a time. If you were to say, come back in a few years and give this talk again, you're probably going to be seeing a lot of this. Now, unclear what is the best combination, but you can sort of imagine in your mind that some of these might be better at barriers, some of these might, this going faster, some of these might be good at itch. And so I'll leave you with that there's so much happening here. I had to even edit stuff out of this as well, but this is going to continue to be a really cool and exciting area. Thank you.
Thank you. That was very fast forward. So, yeah. Who else? We are very privileged to have at the end of this symposium, wrapping all these things up, because we heard that some things are working, some things are not. Maybe we are not doing this, we are not taking the proper angle. And we have to look into a holistic approach, which is called One Health. And this topic will be introduced by Dr. Karina Do, who is the chair of Department of Environmental Health and John Rock Professor of Climate and Population Studies at Harvard Chan School of Public Health. She's also the perfect example of how to combine, research, basic research, clinical care every day, and also public outreach and policy outcome in the implementation, and also being a very empathic person that we love very much. Thank you, Kari, for joining.
Thank you so much, and thank you for those of you who have stayed this long. I'm so grateful to the International Eczema Council for inviting me here today, as well as Yona for organizing all these wonderful speakers. I feel like hopefully what we'll do is wrap up things and then also have our panel. So thank you again for your attention. I want to talk about this One Health approach because our health on this planet is inextricably connected to the planet. And because of that, we know a lot about exposures. That's been talked about today. Exposures to things that are natural and exposures to things that are not natural, and that affects our skin. So I just want to give you the outline, which I'll talk a little bit about the background of why this is so important, and then talk about the One Health perspective, and then also, unfortunately, those exposures leading to atopic dermatitis and mechanisms therein. And then, most importantly, talk about solutions, promoting health and prevention, looking at emollient applications and mechanisms, as well as looking at biodiversification and interventions that are related to microbiota and improvement of function of the skin and the gut to be able to improve the immune system to possibly decrease and prevent, as Wanda was mentioning, atopic dermatitis. And then I'll talk about conclusions. So all of you know this very well. I won't belabor this point. But atopic dermatitis, of course, also indirectly and directly affects how we respond to our overall environment. So the environment affects atopic dermatitis, but vice versa as well. People can't sleep as well. People get other diseases. It costs money. These are stressful issues. So I think it's important to put the whole perspective of any one individual in any disease. I call it the sociome, to understand this disease and how to prevent it. This is a paper that was published recently by our colleagues in Korea, and I note this because as we think about endotypes and phenotypes of disease, as has been done with asthma, we think about this for atopic dermatitis as well. And you can look at these different clusters of typical atopic dermatitis
individuals. Now, this is not perfect for all populations. This is a study that was done in Korea, but I think their approach was very interesting because you break it down and you look at these types of different clusters in atopic dermatitis. You can look at late onset, early onset, and to the degree by which it was associated with outcomes like allergic rhinitis, like wheezing, like food allergy, like Sharon had mentioned. And I think this is helpful to construct better ways to approach atopic dermatitis and how it relates either via the atopic march or being true, and related and associated with diseases that typically allergists are taking care of. So now let's move forward with the One Health approach. As we think about atopic dermatitis and the different clusters that have been found, there is reproducible evidence that the environment plays a very critical role in atopic dermatitis, not just starting atopic dermatitis, but also sustaining it and worsening and exacerbating atopic dermatitis. So this One Health approach to allergic disease, I think, is a concept that we all need to look at carefully because as patients, we take care of allergy patients chronically, atopic dermatitis included, and we need to think about, like Iona had mentioned, holistically, what environment is our patient living in, what contributions of the environment plays a role in their disease, and how can we help that individual better balance their lifestyles to be able to approach and prevent any health issues. So the drivers of a lot of allergic disease, including atopic dermatitis, include outdoor and indoor air pollution. There's unfortunately agricultural and farming practices that also increase allergic symptoms, also AD, and as we learned today, contact dermatitis as well. The outcomes include, especially in climate change and extreme weather conditions, loss of biodiversity, loss of food insecurity, and importantly, epidemic and environmentally driven pandemics across the world. The One Health approach, I think, is really important for our perspectives, that we include not just humans, but also animals and plants. Because again, we live connected to these organisms, and as allergists, we especially understand animals and plants because that affects our patients. So to me, this is really a concept that should be natural to all of us in trying to understand. Why is understanding One Health important? Because in understanding this, we can have transformative outcomes. We, as trusted individuals there for policymakers, for our patients, we can show that by understanding all of these transdisciplinary approaches to improving the environment, improving animal husbandry, improving human health, that will reduce the risk of future pandemics, will reduce the risk of food insecurity, will improve climate mitigation and adaptation capacity. For those of you interested, the conference going on right across the way that many of you might have walked past is actually a solar energy conference and battery storage conference. So I urge you during your breaks, go ahead and go to that conference if you can, because I'm finding out a lot about alternative energy sources and renewable energy right here during our allergy conference. So what are the exposures that are leading to atopic dermatitis and mechanisms? It's important, again, holistically, to look at all of the exposures that can affect a derm. We've spoken a lot about them already, but anything that disrupts the barrier, anything that can lead to Th2 immunity as well as chronic inflammation, when you look at all the different types of blockade mechanisms that could improve atopic dermatitis, that just tells you that upstream, there are a lot of different pathways that could actually cause and activate those targets that are needed to be inhibited as some of the therapy approaches that we just heard about. There's microbial dysbiosis that also I'll talk about as it pertains to atopic dermatitis. So looking at the nonspecific external exposures in AD pathogenesis, stress plays a role. We heard about viruses as well, the triple threat that Wanda mentioned, economic development, migration, urbanization, loss of biodiversity, climate change, and importantly, looking at all of these contributions from the externalities to the cellular and to the molecular basis of these disease. Individual exposures play a critical role. Antenatal exposures, what's been found here, and this was a review that was recently published in Allergy, helminths and farm animals versus antibiotics, early life exposures, emollients, breastfeeding versus Western diet, loss of diversity, leading to this dysbiosis that then unfortunately causes the immune system to be changed, and genetics plays a role in this as well, and then that all plays a role in atopic dermatitis pathogenesis. Going even deeper beyond the skin, looking at different levels of Staph aureus infection. I think you had a wonderful presentation by Anne-Marie earlier today, and looking at the restoration of the cutaneous microbiome is very important. So let's talk about this dysbiosis and microbiome. Endogenous features and exogenous factors affect a person, again, as a whole body. And when we think about atopic dermatitis, a lot of us have already talked about those other allergic diseases that unfortunately are complications of atopic dermatitis as well as allergies. And that dysbiosis is important because it affects the epithelial barrier. That epithelial barrier, anything that touches the air is basically lined by an epithelial surface in our body. And so you have lung, skin, and gut affected by the same compromising dysbiosis that we need to think about as physicians. I'm really glad that Wanda mentioned about some of the bacterial lysates that are being tested now, because some of not just the live bacteria, but also their products can be looked at carefully as to promote skin barrier health. Dr. Rosemarie DeCroix, who's at Stanford, wrote this review a couple years ago, but I think it's a great review to talk about all of these issues holistically. Sorry. So let's talk about solutions. I don't want to be Debbie Downer today. I always like to make sure that in all of my talks we can talk about solutions for change, because we can be implementers of that change as researchers, as physicians, as well as trusted voices to talk to policymakers. And this is a study that is the PRISMA study. It's a flow chart here to show you a systematic review that was done to include studies to test for different emollients. And as you heard from our colleagues, not all emollients are the same. Some are paraffin embedded, some are petroleum based, others are more natural, but we need to be careful because we don't necessarily want to promote an emollient that could induce a contact dermatitis or make atopic dermatitis worse or promote bacterial overgrowth. So on the whole, these meta-analyses that were chosen, 10 studies were looked at carefully, but you can see a reduction on the whole in atopic dermatitis and prevention with emollient use. So like Wanda said, moisturizing is really key to that, but moisturizing with the right emollient is also critical. Not all emollients are the same. So what they found in this article is that prophylactic emollients applied to the first 6 weeks of infancy reduced the development of AD. There was significant benefit where prophylactic emollients were used continuously to the point of AD assessment. But importantly, each emollient might be different, so we need to understand which one is more helpful than the other. So a small study was done at Stanford. Dr. Tina Cinder, Dr. Don Leong, and Dr. Sharon Chandraja are pictured here. What we did back a couple years ago was to look at whether or not a ceramide-based emollient versus a paraffin-based emollient could actually change and be differently therapeutic or preventative for the transepidermal water loss that we were looking at as a tool. So our hypothesis was that the ceramide, the more natural-based trilipid cream, would actually have an improvement in tool compared to, let's say, a paraffin-based emollient. So we looked at only 45 participants. We used their right arm versus their left arm. So it was a controlled study, but the person was their own control, and you can see the demographics there. So what we looked at there is compared to baseline. So even after just 5 weeks of treatment with Epiceram, which is the trilipid I spoke about, the tool went down to normal levels after the Epiceram versus the Aveeno, which in this case went down, but not to normal levels. So I think this is really interesting as we explore which emollients could actually reduce atopic dermatitis and therefore potentially reduce the atopic march proclivity of certain patients to go on to develop other allergies. So with that perspective, Sharon had mentioned this, thanks to the NIH, we now have a funded study where a combination of regular skin emollient use early in life with topical steroids, or what we call proactive care, versus reactive care, which is what is generally done now in populations around the United States, we have comparator arms. And we screen individuals at 0 to 12 weeks of age. They have dry skin at baseline, and then we randomize them to these 3 arms. And we're going to be analyzing this data soon. The trial is still enrolling, but we're really excited about this, and hopefully we'll be able to show decreased food allergy sensitization. I would be remiss if I were not to call out the fact that there are missing individuals from the NIH that are part of this study, and we're so grateful to their help. And Alcas Toyas, Erica Britton, Adora Lynn, Maria Veri, Susan Perry, they're all critical to be able to do this study and to accomplish our goals for SEAL. Inclusive, we have a multinational PI group that is helping with this study as well, from Stanford to Denver to Chicago to Cincinnati, as well as in the UK. So we're really excited about the study. We hope to be able to share results with you. Another solution is biodiversification. And many of you have heard about the animal study. I myself made sure that all my children were exposed to more than 7 animals at once after seeing this study. I wouldn't recommend it for everyone, by the way. It's not easy on cleanup crowd. But importantly is this data, I think, is instrumental to our knowledge of mechanisms. So this was a work that was done by our colleagues in Wisconsin and other groups looking at atopic dermatitis as it relates to reducing the risk of atopic dermatitis with increasing farm animal exposure and increasing diversity of the species of each animal. So that, I think, turned a lot of people on to how do we improve biodiversification, and how do we, again, within that One Health approach, really test greening the planet, going back to basics, and making sure that we get exposures to things that are natural and that were, as we went through evolution as humans, also part of our natural environment that to some degree protected us and our immune system developed because of these features. So this is a great study, again, a small study that was done in Finland, where they looked at the intervention before and after 28 days. So the intervention only happened over 28 days. So they looked at 4 centers in Helsinki, where they enrolled children, and you can look at the age here. These are young children. And they had standard centers that had macadam on the ground. These were typical daycare centers in the city, versus those centers that were more natural or nature centers. And then they had those centers that they actually rebuilt, and they then intervened, and the children went from standard to nature within the period of 28 days. So they looked at microbiota in the skin, and this is important because the diversification was quite different in the skin as well as in the soil. In the intervention group, as well as in the standard group, there was much less in the standard group. In the intervention group, it went up even after 28 days. So what does that mean? Are we improving dysbiosis even within a 28-day period after biodiversification of an environment? So the immune system was also looked at in these children. And what I found really interesting is that the IL-17 was reduced, and that even though, again, it's a very small study, so this has to be repeated, but TGF-beta was improved in the intervention, again, even after 28 days. So in conclusion, this intervention with biodiversification of the environment changed the skin, especially the gamma proteobacterial communities, increased TGF-beta and reduced IL-17, increased IL-10. I didn't show that data. And it suggests that biodiversification might be helpful if in nursery school, but hopefully as we also look at biodiversification of other elements of our environment. So in concluding, in the US, AD affects about 16.5 million adults and about 10 million children. AD increased risk of other allergic diseases, as we've spoken about with the atopic march. Immune dysregulation, barrier dysfunction, alterations in the skin microbiome are key intrinsic factors and can be influenced by both genomic, epigenomic, and environmental factors. Use of emollients to maintain a healthy skin barrier has been implicated in reducing risk of AD, and that continues to be studied, and now and in the future. An NIH-funded multinational, multi-site randomized prospective interventional study called the SEAL study is currently ongoing to test whether emollient use will reduce food allergy sensitization. And then exposure to biodiverse environments such as pets, farm animals, and rural environment could perhaps assist in building immune tolerance and reducing atopic dermatitis. So thank you for your time, and I look forward to questions.
I would like to invite all the panelists to join me on the stage for the discussion and questions. Please join to the stage so that we can go ahead with the discussions. I will encourage the audience to go to the microphones and come with questions. I will start with the ones that we have being addressed online. So, go ahead. Yeah, absolutely. So no, please go ahead. I'll just stand here. So yes, absolutely. Sharon, please go ahead.
I know.
That's okay. It's a great position to be in.
Okay, so the first question goes to our first speaker, Dr. Zelma Chiesa, and the question is about, with the push of the labeling for penicillin for pregnant women, what are we expecting in terms of prescribing antibiotics and, decreasing the risk of AD?
So I think the question is about prescribing penicillin.
No, it's the labeling penicillin allergic women, and then they will receive, proper antibiotics because they are delabeled. And do you think?
Right. I think
because you showed that penicillin was the highest risk.
Yes.
The labeling and this. I think that's the sense of the question.
Right. And I think we have to be, again, careful how we interpret the data that we looked at so far. I think penicillin tends to be a frequently prescribed antibiotic during pregnancy and also early in life. I don't think that based on the data that we have so far, that we would sort of advocate in terms of changing prescribing practices. I don't think the data is there yet. So I don't think it does. I think at this point, we have to be cognizant that we're really beginning, and not to make any puns, but we're scratching the surface in terms of how antibiotics seem to be influencing this risk. So I wouldn't advocate based on what we've observed to change how we treat our patients, including our penicillin allergic patients. I would say that I would probably be more cautious, right? And sort of maybe keeping a closer eye on these patients and their children for future development of atopic dermatitis.
Okay.
Thank you for the question.
Thank you. Now, to Anne-Marie, if we were about to build a risk profile out of your research, what would be your, construct there? And try to do it, in terms of what is helpful in the clinic, So what we can immediately use.
So I think the question is risk for what, right? So if we want to look at risk for AD or no AD, the risk profile was Black children, more recent birth, meaning, the more recent decade of birth, male sex, and then maternal history of either atopic derm or food allergy, no pets. So I think that that to me would be the biggest risk factor, and I think that was based basically on those conditional inference trees that I talked about on there. So that's the risk for AD versus not. If you want to think about the risk for AD phenotype, so who's going to have that persistent AD that's most likely to develop, asthma and the other atopic diseases. I think the risk factors are a little bit different. So basically, we know for persistent AD, again, it would be now instead it's a general risk person, so not high risk. So born general risk, again, Black children are higher risk for that. Antibiotic use was in there, first year of life, and then again, no pets. So I think that would be kind of how I would do that with a risk profile. And then for a bonus, if I'm able to skin swab them, that would be a huge marker based on the data that I showed, depending on strains of Staph aureus, but also the other staph species that I talked about.
Thank you for adding the bonus because I was going to challenge you on the skin thing.
Yes.
Okay, so for Sharon, omalizumab or dupilumab? And if you have, let's say, a profile for choosing either.
For what? For treatment of what?
For prevention, we are discussing. For prevention. Yeah.
Oh, for prevention.
Yeah. It's even tougher. So.
I know. Let's ask.
Dupy wasn't around when I started.
No, I know. I think that's really hard. I think we see that dupilumab is a great drug for treating atopic dermatitis. And in reducing IgE, those studies that have followed the trajectory of specific IgE, I was trying to do the calculations in my brain, 156 weeks, 3 years, really to get the 90% reduction. As Wanda said, IgE is the driver of atopy, so we think, right? And omalizumab is a great reductor of IgE, but we see variability of response in asthmatics, in food allergic individuals. And so if we had a perfect anti-IgE reducer, I think that would be the choice. But if you had to pick one, it depends on duration of treatment. So can you start early? Can you treat long? How long do you need to treat with either? To be able to change the immune system? I think we don't really know yet.
Okay, Wanda, would you like?
I think this is let's find out the results, right? What happened because the anti-IgE. One other thing that omalizumab, it works for food allergy. It also, there's data that it restores these interferon, alpha interferon and antiviral responses. Dupi may as well. I think if PARC doesn't work at all, then we'll have some other enigmas to think about. If it does, then we kind of know, at least along that pathway. If you could do it both, the only thing would be nice if you could have a mix of those mechanisms and something that's not a shot would be nice for prevention too.
Or maybe
maybe developing small molecules or something along this pathway.
Or maybe we could have a responder profile for one and a responder profile for the other, and then we do, select it.
Yes. But in all fairness, when I started thinking about PARC when I was an assistant professor, like years ago, and to get the FDA to allow us to do this and all these things, Dupi was a concept. And then it quickly got approved, and there have been discussions on doing it, but they have not. They're not going to be done. In other words, we just know they're not going to be done. But we're going to learn a lot, and doing placebo-controlled studies is so challenging, as you know, for this age range. But you got to do it to answer the question. If we just do willy-nilly, we won't answer the question. So, exciting times.
Yeah, we're all waiting for Wanda's study.
Absolutely, both for Amber and Marcella. So, is there a special risk profile for getting this contact dermatitis? Again, advice for clinicians. What would you look upon, in building this risk profile? So it depends, for example, on the toxicity on the epithelial barrier, or it doesn't. How do you see that? It's substance dependent or person dependent, or both?
You mean a risk profile for developing
yes.
allergic contact dermatitis in atopic dermatitis patients?
Yes, or irritant contact.
I hope you're not asking for a genetic risk profile because
No,
I can't answer that
absolutely not.
question. I'm a clinician.
I'm asking for something valuable for the clinician.
So, in atopic, I mean, I think we talked about it. It's the barrier is the risk profile. So we know that the barrier is not normal for both lesional and non-lesional skin. So literally all patients are at risk if they're applying products. In theory, right? We talked about there's 2 reasons why they wouldn't be at risk, and there's 4 reasons why they would. So they're all going to be at risk. I don't know if there's a specific profile. Maybe the heavy applicators, people who are applying tons of products all the time. Honestly, one of the individuals that did one of the emollient studies in atopics early on told me about his study, and I was like, you can't put all those chemicals on those babies, right? And I'm still paranoid about that this frequent application of product, even as a prevention, is a risk. Like, Yeah, that's a contact dermatitis specialist perspective, though. But in my opinion, anybody who's applying a lot of products is at risk.
Okay, that's fair enough. Okay.
Clinically, I would say, you have to have a high index of suspicion. So you have to think about it. And I think a lot of patients may be underdiagnosed because allergic contact dermatitis is not considered in patients. So have a high index of suspicion. Amber talked about, new areas dermatitis in patient who has a classical picture. So facial dermatitis in someone, new hand dermatitis, multiple products that have fragrance, particularly a lot of the preservatives, MCI, MI are big contact allergens. But I think just clinically suspecting it and then patch testing your patients is the most important thing.
Okay, great. Thank you.
Yes, thank you very much. Thank you for your conferences, your excellent information. I would like to ask, what's your opinion about allergen immunotherapy alone or combined with dupilumab, maybe, as the prevention of allergic march? And another question about the nanobodies that are maybe the newest, like lunsekimig, that combines 2 targets in TSLP, interleukin-13, in different epitopes.
Okay, so who wants to take the allergen immunotherapy one? Yeah, I have for him. But
Well, you guys parse that out. I can work on the second question for you. So it's a great question. And I think that there was just so much hope behind tezepelumab as like an individual therapy for atopic dermatitis. And there's a lot of reasons why maybe just targeting a singular alarmin just wouldn't work. Maybe it's a matter of finding the right time to do so. Maybe just that by itself is not nearly enough considering how many signals do that. So there isn't data for me to say that that approach is going to work, but I think that probably the best design bispecifics or trispecifics are ones that are probably taking something that we know, like a backbone of type 2 inflammation, ones that are probably taking something from a keratinocyte alarmin level, and then potentially IL-31 seems like almost the perfect combination to add to something too, just given how rapid it is, particularly for itch resolution and directly acting on neurons itself. And you kind of have designed a perfect 2 or 3 part drug. But I think that most of our upstream targets will probably be much better in efficacy when combined with something else.
Okay. Thank you very much.
and for allergen immunotherapy?
I mean, I can try to tackle that. So there have been some studies that show that's promising. It's a different approach than using like Xolair, which blocks the response to exposure. It has it's like big guns, right? Blocks all of IgE. While allergen-specific immunotherapy is actually introducing kind of like the Gideon lac and introducing early peanut was preventive. So you're kind of asking about giving it would not be easy to do shots and the Dupy but you're kind of doing both approaches because Dupy we know reduces IgE. Dupy, or if you use Xolair, reduces. Then you're introducing through the immunotherapy. We have a study that was not funded through NIH, but in the UK there is now a dust mite slit prevention study that was finally just funded. Hassan Arshad, who might be at this meeting coming from the World Allergy Organization, is launching that study. So we'll learn that might be a possible strategy, and it's also sublingual. It can be given orally, but it's it's targeting just a specific phenotype. The thing about the biologics is it gets a whole pathway. The allergen-specific immunotherapy is introducing early, just like Gideon Lack did the peanut early. And so they're different approaches. We might need a combination, but also practicality and what may work is still in question. But great question.
So I can't talk about in terms of prevention, but in terms of treatment of atopic, dermatitis with either subcutaneous immunotherapy or sublingual immunotherapy for dust mite, the literature is a little bit mixed on this. It's not a strong signal. Some studies show that it improves the atopic dermatitis. Some shows it makes the atopic dermatitis worse. So the literature is not really clear in terms of treatment of AD with immunotherapy.
Okay, great. Sharon, anything to add on that? No.
Yeah, I mean, I think combination approaches are really exciting to think about when you're thinking about beyond AD, further along in the atopic march, because it's about making it more tolerable for the patient, allergen specific, which has the potential for long durability. These other biologics are kind of a Band-Aid, dampening down allergic inflammation, but not really stopping it so far.
I was not involved with the study, but there is a published study looking at tezepelumab plus a cat skit, alone or together in each. And it did show that the combination had higher efficacy than either alone. So those patients, it was more focused on asthma, not AD, but in terms of your question with regards to the atopic march and how combination could be.
Okay.
It's just a really quick question. First of all, it's such a pleasure to have so many experts in the room on eczema with so many different areas of interest. So thanks for all your lectures today. I'd like to back up a little bit because as an allergist, the history I get over and over and over, and this is not everybody, is delivered, mom is allergic, that, or their family members that are allergic, delivers by cesarean section, doesn't breastfeed, and then boom, here we go. And it makes me wonder, what should so I had a granddaughter and I said, listen, if there's a C-section, this is what I want you to do. And they said, you are crazy. You're not coming near the delivery room. But I just wondered if you have any thoughts about, what should we be doing? Is there a microbiome that we know of that we think is non-allergic that we might be introducing to the kids early on rather than maternal flora if mom's already allergic? Or should we go with maternal flora because that's what we have and that's what's in the environment? That's kind of the general gist. Should we change our approach to these kids since it happens over and over? Thank you.
Thank you. Thank you for the question. Now, for Raj, a very quick question, because you mentioned a lot, of interventions that are potentially there, but looking at all these trials, there is a high percentage of non-responders. Even if you go higher and higher and higher, they're still non-responders. Where do you go from here? How do you manage that? And related to that, there was a question on the high placebo rate in all this. So please try to tackle both.
Right. Okay. Well, let's fix everything. So, we can touch on the latter part first. And in the interest of time, I'll keep it to very brief what my thoughts are on that topic. So we've seen an explosion in studies in atopic dermatitis, ones that have patients that have to fit certain criteria and a lot of pressures for getting these patients in, and perhaps a lot more sites than there used to be when there weren't that many atopic dermatitis patients. You can kind of see where I'm going with this. So there probably is a lot more heterogeneity in measurement that's happening in a lot of these trials than maybe we used to see in some of our trials previously, and a lot of people that are probably less severe than perhaps the criteria might want the patients to be. And thus, that's probably one reason why we're starting to see placebo rates that are fluctuating a little bit more than previously, because we actually have fairly valid tools for doing this kind of stuff. So that's one reason, but there's probably others. The question of non-responders is a fascinating one, right? And there's nobody that has billions of dollars from the pharmaceutical side that usually wants to spend that much time studying those non-responders, even though it's brought up every single time. And so the hope is that with some of these broader but more selective targets, maybe that population decreases, but you're correct, there always appears to be sort of a group of people. What would be fascinating is, are those people actually just not responsive to everything, or is it just very specific modalities as well, which is one. Two, is it just simply a matter of metabolism and the way that they handle a drug, or is it truly mechanistic? Three, we don't understand genetic polymorphisms in this space, right? But we know for small molecules, let's say, just a simple alteration, sort of a JAK inhibitor in a small molecule and a kinase site can have a profound effect on whether or not that's truly going to bind as well. So is this indicative of now the genetic layer of sort of the complex polygeny that underlies atopic dermatitis? Question mark for all of those, but that's all possible.
Okay. Thank you.
Can I add to that? I seriously wonder whether some of the non-responders might not have the disease that we are trying to treat. So atopic dermatitis historically is clinically diagnosed. And as someone who sees dermatitis all the time, I'll tell you there's a lot of misdiagnosis by both dermatologists and other types of specialists. And to the first question that Raj answered, if you really look at the severity of published trials, Raj mentioned maybe they're not as severe as we are saying they are. But really, when you look at the data, you can see that the severity is actually reported as less severe over time. So the very first Dupy trials, patients were more severe than the more recent studies that we're seeing on paper. It's not just, they're just not as severe as we think, but they actually aren't as severe over time because we have so many more patients out there that are being studied.
Okay. Last two questions for Kari. So, nature-based solution, fine. But we need to make sure that the nature that the patient is exposed is healthy and not polluted. Huh?
I'm going to put my air pollution and climate hat on now. It's critical that we start mitigating and reducing air pollution and using filters, adapting to air pollution as well, not only individually with masks for our patients, but also importantly, air filters in rooms, and Wanda's doing a lot of research in this. So reducing air pollution, reducing PM 2.5 is critical to all of this. Using renewable energy sources, decreasing air pollution and mitigating against the use of polluted products is very important. The petrochemicals that are used, plastics, we don't know how microplastics and nanoplastics affect the skin, but it's hard to believe that they don't. And a lot of these emollients include nanoparticles and other particles that could embed in the skin and be problematic. The last piece to this is the fact that we evolved with nature, with this biodiversity, with animals, with plants. And of course, we as allergists, we worry about pollens and plants, but there's a way to use ecologically friendly plants as well as animals to be able to improve the health of our patients. And I think in this overall message that we're talking about in this council and in this meeting, trying to make sure that we promote health. We talk a lot about what things can worsen health. That includes air pollution, that includes avoiding wildfire smoke, that includes avoiding floods if you can, but these things are happening more and more. And so as we think about how to get ahead of this and reduce climate change, we need to be part of that solution. Reuse renewable energy, use less plastics. But we also need to get in better touch with this One Health approach and understand how biodiversification can improve health and reduce disease. But also we have to care about stress and mental health, and that's also a component to being nature-based. Thanks.
Thank you. And both for you and Wanda, how sustainable are all these prevention trials in terms of health economics? Because you will be challenged with that, and really linked to that because I love your point of view with these transformative outcomes. When we will see them integrated into clinical trials, because this will really move the field ahead.
Yeah, so this is always the big question, right? If it works, because these biologics are crazy expensive, right? But I think the approach is that if the concept works, other things may be developed that maybe are not monthly injections, small molecules. That area will open a whole line of investigation. That's why we still need NIH. We still need the partnerships. But then also many of these things may come down in price as well, economics. If we know that insurance companies realize that you treat somebody with something early on for 1 or 2 years or early on, and it prevents a lifetime of disease, that's a health economic win, right? So I think that's why prevention, for those of us who see kids early on, trying to find solutions in this way, can really have a lasting impact from an economic, but also a health and future generations impact as well.
Okay, Kari?
Yeah, lastly, in general, the advances that have been made in public health for prevention far outweigh the risks of having disease and the cost of that disease. So there was a study that was done in New England, again, a small portion of the world, but for every dollar that you put into prevention, you're able to get out at least $6 in preventing chronic children's diseases, and that's just in children. So I think on that economic analysis, we need to think about this. We need to think about reducing costs, but getting the biggest bang for our buck. It's hard enough to be able to convince someone to take an emollient every day for a long time. How do you sustain that? How do you get people to engage in that cooperative experience? But if we can prevent further upstream, I think it's worth it because, of course, we will still have jobs, but it's really great to be able to save on the overall money and for our children too.
Okay, with that in mind, I would like to thank the panel who really made our 10th anniversary for the International Eczema Council a celebration. Thank you all the audience that was until the end here with us. And let's hope that we will meet again in 2 years and then we will continue our discussion. Maybe some results of the trials will be there and then we can provide new data for our patients and make it better life for them. Thank you so much for joining us today.